Also, Professor, so my name is Xiaoling. I will be the host for this session. And if you have any, like if you need any help, you can let me know.
Should we start now or just wait for a few minutes?
Sure, sure. I think we could wait a few minutes.
You're recording now, or?
Yes, it is automatically recorded.
Okay. Could we start off?
Yes, yes, please go ahead.
Yeah, good morning, everyone. Thank you for participating in this short course on the statistical learning methods for neuroimaging data analysis. And because we only have one day, I kind of cover all the materials I'm interested. And also, I'm writing a book and also writing review papers on this topic. And I'm going to send you guys the review papers maybe 2 or 3 months later. But there's something I'm obligated to finish it ASAP. And the first lecture is about introduction. Let me go ahead. And actually, for each of these lectures, I listed some of the references. If I'm interested, you can read some of them. Okay. In particular, some review papers you may want to read more carefully. And the first part is about large-scale imaging-related studies. And I think when I started my career almost 20 years ago, and there are not too many large-scale neuroimaging imaging-related studies. But right now there are too many, okay, in a certain degree, okay. But I still not enough, and you will see that why that's the case. But I think that currently there are 4 major imaging-related studies. One is ADNI, Alzheimer's Disease Neuroimaging Initiative. Second one is a Human Connectome Project. Okay, now it's not a single project, it's projects. The third one is UK Biobank. The fourth one is adolescents brain cognitive development. These 4 studies are most popular standard studies these days, okay? If I'm working on your imaging related to the projects, I think these days you have to deal with them right now. And also there's some other imaging related studies like the cancer genomic atlas But there's many about the cancer genomics, but they also have imaging component inside. And also they have the national long-screen trials and prostate, lung, colorectal, and ovarian cancer, the screen trials. All these three studies I just listed as an example. And all these three studies contain the imaging data, okay? And I think for throughout the talk, I will cover some of these studies. The first one is adenine. Adenine started from the 2004 until right now. They have 4 phases. Adenine 1, 2, go, and 3. Okay, there's the 4 phases. And these studies is kind of one of the pioneer kind of neuroimaging related studies, and in many aspects. Okay, and this is the one thing is because they try to use the imaging as a candidate biomarker for Alzheimer related trials, and also they connect all kinds of information in the studies. These are longitudinal studies. They connect all kinds of AD related biomarkers and also cognitive functions. Okay, this is one of the graduate studies in the history. And the second one, it also make a big impact in the neuroimaging field, is the human connection projects. Okay, and originally they started with a single project is more adult. Okay, and from the 25 to 35, something like that. And the purpose is to try to map the human brain by connecting the structures to function and behavior. The most important achievement for this study is basically develop these new kinds of imaging analysis and also imaging acquisition protocols. Okay, and so that basically NIH basically extended these kinds of the pipeline to the other kinds of studies like healthy adult connections and lifespan connection data. Basically, they try to cover from 0 to 90 years old. There's multiple studies going on with this data. And another thing is basically try to extend the HCP protocols to the connection related to human disease, in particular like Alzheimer, like the psychiatric disease. Okay, that's a very important kind of achievement. And another very important data set studies is UK Biobank. There's one of the greatest studies, medical studies in the world. Okay, and in a sense, they have 500,000 of subjects, almost around 500,000 of subjects participants in these studies. They cover all kinds of data you think about it. Okay, imaging is only small component in these studies because they plan to cover about 100,000 subjects with all kinds of MRI images, okay, in the body. It's not about the neural images, but they cover other organs in the body, okay. And also, this is the largest studies in the world, and you can imagine that the number of the subjects required. They are going to require in these studies is way larger than all these neuroimaging studies in the world, okay? And using the same protocols across multiple sites, okay? And also they have about around 10,000 subjects with repeated scans, okay? Because of this, they will change the whole areas a lot. Because the whole neuroimaging field will be dramatically changed because of this data set, okay? And these days, if you don't analyze the UK Biobank, basically you fall out of the mainstream kinds of neuroimaging data analysis. And another thing related is adolescent brain cognitive development is run by UCSD. The ABCD study is the largest long-term study of the brain development and the child healthy in the United States. Okay, this is cover in most of adolescents, but this UK Biobank is many. It's about 45 to 55 to 65 and older. But HCP these days, because the original HCP is around 25 to 35, and these days they try to expand it. But it takes years to get to really get the data and share it with the community. But Alan is many about relative older patient subjects. This is just to give you big ideas about neuroimaging data you can work on. Okay. And but for most of my papers these days, I use all these 4 different the data set from all these 4 different studies. Any questions? And the second part is what kind of scientific questions or clinical questions people care about. Okay, why should we use the neuroimaging data in our studies or in our clinics? And there's a paper published last month, and it's called the Brain Charts for Human Life, SPECT. Okay, and this group of the researchers did an amazing job. Is what they did is they tried to kind of, it's not connected, basically assemble around 100 different studies across the world together. They covered basically from 0 to kind of 90 years old. They even include a set of subjects is preterm babies, okay? And basically for all this data, it's not uniform, okay, because they covered over years to get this kind of, for this 100 different studies, there's a lot of heterogeneity inside. But in any case, they did this work, and basically they just ran very simple image analysis by extracting the whole brain volume white matter gray matters, okay? And then you basically they create these kinds of quantile kinds of curves, trajectories for these measurements. But I think that this is one of the greatest achievements in the history of the neuroimaging related studies, because it's the first time we are able to see kinds of the whole big pictures about how does the brain develop from preterms until to 90 years old around. Okay. And in any case, this effort is not because of the data, how fancy the data analysis is. It's because of all the effort they spend on by basically assembling all this valuable data set. Okay. And I think if you want to get to the data set, you have to do the similar work as they did. Basically, they won't be able to share the data with you, but you have to do that. Most of the data they are using are public available, but it takes a little while to do all the paperwork. And also the NIH is doing these human collectome, lifespan collectome studies data set. They cover all multiple kinds of ongoing studies. And even the new HBCP studies, they just started from this year, and actually they will last for 10 years. Okay, they followed all the subjects, babies from 0 to 10 years old. And actually, this is one of the great things they wanted to do is try to map the brain. They want to define, they wanted to understand what is a healthy brain. Okay, what does the healthy brain looks like? Okay. And that's very important questions for any kinds of brain-related disease. And this is not just about the brain structures, also people care about the brain functions. How does the brain functions develop from the preterm until to 90 years old? In what sense? Okay, that's the big, very long kind of the efforts for the scientific communities. If you really want to work on it, this is one of the great questions people are working on. And why do we care about brain imaging for brain disorders? And usually we are using the brain imaging to capture the brain structure and the functions changes associated with major brain-related disorders and the normal development. Okay, when you can define what is the healthy brain is, the next question is you can really think about compare with these kinds of normal chart. Okay, and what is the chart for well, if you got this kind of the brain-related disease, what kinds of the growth patterns you could have, okay? And it's more like references, because normal development is more like references. You can use all kinds of comparisons. And there are all kinds of different brain-related disorders, like neuropsychiatric disorders and neuro kinds of degenerative disorders, in particularly Alzheimer's disease, okay? And actually, Alzheimer's disease is one of the hottest topic nowadays in the science, not just the medical science in general. Okay. And one thing is people will ask me what I'm doing in the last several years in my career. And actually, I have been thinking about why should I do this neuroimaging related research, in what sense? Okay, I think many people know that I have been working on combine the genetics or genomics information with the imaging information together. And this is basically the past diagrams I have been thinking in my mind all the time these days. And you can think about this, we start from genes to the brain disorders. And actually, this is not just, this is only one big pictures from gene and basically to the brain disorder. Actually, they go through to these kinds of molecules, cells, brains to brain disorders. Okay, they are highly related to each other. In a certain degree, this is basically the causal pathways going through that. Okay. And also all these things, these kinds of the pathways also confounded by environmental, social, and the psychological factors. Okay, and all these pathways are confounded with that. That's the reason, nutrition and also other environmental factors are critically important confounding factors for these pathways. And then we are interested in how to build this up. Okay, really try to understand and how the different components for these kinds of causing this kind of brain disorders in what degree. Okay, there's something that's what I'm working on called the brain imaging genetics paradigms. Okay, that's the big pictures you have. The one thing that we are working on is basically the brain charts for human lifespans. And also we wanted to try to really integrate imaging with all other type of data together. Okay, that's important. And also all these things is also related to other type of data, in particular when working on the disease related to the problems. Actually, one is related to EHR, electoral health record. And that's one of the reasons we are doing these kind of things these days, basically, how to really handle the EHR data, right? Another thing is basically related to precision medicine. Regardless of, for all these different disease, and we are researchers we are interested in precision medicine. Basically, we try to develop this personalized precision and health management for that. And one great thing in the last 10 years is we have all kinds of tools and different devices, and also we have a lot of development in hardware and the software. Nowadays, it's much easier to record the changes, all the vital signals, signs of individual from birth to death. Okay. And including your personal living habit, past medical history, diagnosis, treatment, all of these different things, and including imaging data. Okay, you think about it. And these days, compared with 10 years ago and even 5 years ago, I think these days you have for the students in the statistical, biostatistics, and also any field in the data science. Okay. And if we are working on the medical imaging, related to the studies, you have to deal with the data across the different domains. Okay, you have the clinical or behavioral data, and also you have all kinds of imaging data, genomics, genetics data, and also all kinds of environmental kinds of data. Then you can think about how to really link all these things together in what sense, okay? And these days it's very important to make your hand dirty, not just stay at home and kind of don't just to do some theory. That's not the current data science. And if you work on the clinical questions and regarding to working on the cancer or the brain-related disease, usually the imaging data is a very important component and or tools for the scientists or physicians to solve the full level of questions. The first one is basically people can do prevention, okay, and that's commonly people use in particular. And people are used in the like a cancer, for breast cancer, we do CTs, okay? And also do diagnosis is very common people used, and also treatment, and also the prognosis. And this is basically the imaging is in particularly popular for the diagnosis and the prognosis, okay? And that's the most popular things people are used in practice, okay? In current clinic setting. And you think about why we care about imaging data. Imaging is just a kind of a device to take the pictures, okay? And you can take the pictures in generally for a specific organ, like your brain or your heart or other kidney, all these things. And actually the different imaging modality, just different kinds of devices that help you to take a picture about the organ's function and the structure, okay? And for clinical questions, we are basically your kind of, when you have the imaging devices, they allow you to sequentially or longitudinally to follow up all these subjects, okay? Ideally case, you're supposed from the beginning to the end, okay? Then you can follow this subject for a long time so that you can see how does the organ changes across time in terms of both function and the structure. And in practice, we basically have all these kinds of these four major type of the data type, okay, across different domains. We are basically try to use them or use part of that to address the four level questions related to disease. That's the reason it's very critically important in practice, okay? And one of the questions is applied to all the students from statistical or biostatistics. Are you ready for big data era or not? Okay, this is very important. Statistics is not about to prove another central theories. I think the most we are pretty good at that. But I think that for next generation students in our your statistics and biostatistics, you really need to think about how really play with the data. And you need to know how to really process the data and really know how to manage the data, okay? That's super important. And also integrate the data, different data types together to address the scientific questions or clinical questions. This is very important, okay? Any questions? Yeah, if you have any questions, you can interrupt me any time. Don't feel, yeah. And the next question is basically the part is about imaging science, okay? And actually, what is the imaging science? There's no, you know, it's not a well-defined terms. It's basically in Wikipedia, that's what I copied from that, okay? This imaging science is a multidisciplinary field concerned with the generation, connection, duplication, analysis, and modification and visualization of images, okay? That's it, okay? And actually it's an evolving field, including research and researchers from all kinds of different fields, okay? You can think about it. If you want to use the imaging devices to connecting some data, or you want to develop some new imaging devices for scientific research or clinical purpose. Okay. And actually for the imaging science, there are three major components. The first one is image acquisition. Okay. And basically they try to study the physical and mechanisms and mathematical models, algorithms by which the imaging device generates imaging observations. The second one is about imaging interpretation and application. Is to see and try monitor and interpret the target work patterns being imaged. The third one in imaging process is basically any linear or nonlinear operators that operate on the images that produce the targeted patterns. So that's the for relate to imaging science. Okay, but we are not just working on the imaging side. Only imaging is one key tools or components or the data type for our kind of research. Okay, if you're working on the medical research, actually you are working on more general, more broader than the imaging science. Okay. And what is imaging from mathematical perspective is basically imaging is a function. Basically you have these kinds of x tilde defined in omega in the k space. Okay, this is the k-dimensional space. Then you observe a point observations and for that. Okay, omega is Uranus compact set. Uranus will also require additional assumption conditions to impose on it. Okay, so that basically the images we are looking at is have very kinds of concrete structures inside. Okay, like these tiles, they have very strong piecewise smoothness property inside. Okay, you'll see these kinds of the pattern. And also in practice, you need to remember that all the images we're dealing with, most of the image data we're dealing with is discretized image data. Okay, this is discretized in the two aspects. One is basically the omega, the domains is discretized. Another thing is your response F also is discretized. Okay, and all these current devices, the digitized imaging data is always written like F is omega 0 to the kinds of digitized data, the observations. And in general, this is basically the several questions when you're looking at the imaging data. They have these kinds of sampling, the grid points. Okay, for all the points in omega 0. There's two important concepts inside. One is the sampling rate. Okay, that's important. That's the relate to your spatial resolution. Sometimes also relate to your temporal resolutions. And also this quantization is also important. And basically, and this is basically you also wanted to downsample the observed F. Okay, and you want to really record the continuous variables that much. Okay, and in general, general digital images is defined by these general functions. Okay, the F, X, Y, Z, T, S. Okay, that's basically the typical domains you're looking at, and also these quantizations, the scales, the resolution of the quantizations. X, Y, Z Uranus is related to spatial parameters. T is related to time. S Uranus is spectrum parameters. Therefore, cover majority of this kind of image modalities. Okay, and this is basically that's unified kinds of definition of the imaging data or digitized imaging data. Okay, and this is basically the relate to that. And the first part we are talking about the imaging acquisitions. I will go through very quickly. I just give you objectives. You know, if you want to really know all these detail, some detailed information about the different image modalities, you have to go to the literature to find out. Okay, and the most things we are dealing with here is medical image. And basically, the medical imaging is a technical process used to create images of human bodies. Okay, and for the clinical purposes of medical science. Okay, and actually, this medical image become more and more popular these days. Okay, you will see this kind of data, and all the times. in different studies, okay? And this has become much more common than before, okay? And that's the reason we need students to really know how to process this type of data, okay? That's super important. And most of the common, one of the popular things is in relate to X-ray. And this is basically the key procedures of how do you generate the X-rays. X-rays is ionizing the waves considered on the photons to travel at the speed of light with this energy. And you'll see these kinds of pictures like that. It's very common. X-ray is very common for like dental research and also dental clinics. And also when you go to the airport, and you know, you always see these kinds of X-ray kinds of machines in the entrance of the airport, okay, for security purpose. And actually in clinics, the most popular clinical imaging model is the CT scans, okay? And the computed tomography. The CT scans is an imaging method, and they employing this tomography that created by computers. It's basically they use to generate a 3D images of inside object from a series of the 2D X-rays taken around a single axis of the rotations. That's basically the most commonly used the kinds of the image model is the CT. Because in particular, if you're working on the cancer, the number one image type is the CTs, image model in clinical aspect, okay? And it's very sensitive to some of these components in the tumor. Okay? That's the reason CT is the more popular in clinical aspect, okay? And another things you relate to PET, okay? This is basically the pictures of the kinds of the PET. Basically, you choose one specific type of the tracers, okay? Then you inject to the human bodies. And then you basically, then you put a subject in the PET scans. And basically then PET scans to try to measure the number of the components of these kinds of tracers in different locations. Okay. And this is called PET. This is a functional imaging techniques. They extensively study the relationship between energy consumptions and neuronal activities. Okay, and basically they use PET radioactive tracers that attach the molecules that enter biological pathway of interest. Okay, and this is a very important tools, but usually it's not a very because it's in certain degrees invasive. Not even though the side effects are not great, but it is still, you know, the prevented from using PET for like neonatal studies. Okay, and this is the basic FDG is one of the most popular kinds of PET tracers people used. Okay, and PET is different from like CT/MRI or ultrasounds because the PET is many and they can provide physiological and molecular information of the brain, okay? And this is very valuable tools, okay? And it's clinically indicated for pre-surgical localization of the seizure origin. They are covered by most insurance providers. The PET is mainly for working for these subjects that has like a stroke or other kinds of the damages. And another thing is ultrasound. Ultrasound is ultrasound imaging involves the exposure part of the body to high frequency sound waves to produce the picture of the inside of the body. Okay, it's cheap. Okay, it's easy to do. Okay, but it's not. You know, this is because of these the technology limitations. And the data is pretty noisy in certain degree, okay? But this is just to show you that this the ultrasound has been used in clinics. And if you do, this is a basic ultrasound image of the photons, okay? And you can look at the kinds of these ultrasound images. It's they yeah, the ultrasound has been used to the clinics a lot, but not that compared with MRI and the CTs. Okay, and MRI images and MRI imaging. Okay, MRI is visualizing the detailed internal structures. Okay, and the good contrast that provided between the different soft tissue of the bodies make it useful in brain, muscle, heart, and cancers. And also, they doesn't involve any kinds of radiation. Okay, and the idea is very simple. They just use a powerful magnetic field to align the magnetization of some atoms in the body. Okay, then use the radio frequency field to systematically alter alignment of these magnetizations, and this basically creates the images, the contrast. Okay, keep in mind, MRI is not measure. MRI is trying to measure the contrast. Okay, it's not a measure absolute values. Okay, the usually this you see the pictures in the MRI images. That's basically the differences of the sum of the tissues. Okay, and actually the MRI these two the Paul and Peters was awarded 2003 the Nobel Prize in Physiology and Medicine for discoveries concerning MRI images. Okay, MRI has become the very important clinical diagnosis and tools, and also research tools. And this is the typically MRI T1 images, and you see these kinds of the brain, and also all these cortical, subcortical regions inside the brain. Okay, and you will see these kinds of the brain anatomies. And another thing is related to the functional MRI. Then that's this one we mainly talking about. People also call it structural MRI. Okay, and this is just they try to map the brain anatomies. But the functional MRI to try to measure hemodynamic responses. Essentially, the idea is the change in blood flow related to neuronal activities in the brain or spinal cord of the humans or other animals. Okay, and actually since the early 1990s, functional MRI has come to dominate the whole brain imaging field. Many due to the non-invasive absence of radiation exposure and the relatively wide availability. Okay, yes, you think about it, the MRI scans nowadays is you can find in most of larger hospitals. Okay, okay, it's very standard kinds of tools. Nowadays, even they try to build the MRI scanners in the car. Okay, and for and then basically you don't need to go to the hospital, and you can just do the company will send the kinds of the MRI machine to your home, so that you can do the scanner in a vehicle. That's the reason what I said is you will see more and more this type of MRI related images. And this is basically there are several different type of the function MRI images. One is a task related function MRI, another one resting state function MRI. And for function MRI data, in each of locations, you will see these kinds of one-time series. Okay, you see the basic is you are working on time series. That's basically one time series at one location. Okay, and this is one function MRI. Another thing is related to the diffusion weighted tensor MRI images, and also we call the diffusion weighted MRI. This can provide information about the damage to parts of our nervous systems and about wide matter connections among the different brain regions. Okay, and basically you have this the white matter is basically these kinds of all these things is white matter bundles. Okay, they connect the different regions together. Okay, and actually, and the brain signals basically can transfer across the different regions, primarily due to this. And the diffusion weighted of these things is mainly related to, for function of mind, we're usually talking about the gray matters, okay? And this is, you see that this gray part of the gray matters is cortex plus nucleus, is basic cell bodies, okay? If you're looking at these neurons, okay? And these kinds of the cell bodies, okay? This is dendrite, all these things. And this is basically is mainly in the gray matters. The white matters is all these axons, okay? And this is basically the white matters. And also you have these kinds of signal directions. This is basically, essentially, the dendrites try to connect the signals from the outside. And basically they transfer signal through these kinds of axons or mining sheet. And to these kinds of when they have all these signals, then transfer to your bodies, the different organs, okay? And basically the mining sheet speeds the signal conductors, and basically the axon prizing sheet together equal to neurofibers, okay? The diffusion weighted images, you mainly try to reconstruct these axon and plus mining sheet called the neurofiber structures, okay? Then they try to recover this part. Okay. The function of my mainly basically try to see how does the signals transfer and across different regions, brain regions, and also organs. Okay. The major white matter pathways aggregate many fibers into the bundles. Okay. This is the basic about. Here is one typical example, several popular examples, white matter bundles. Okay, you can take a look at the location of these major fiber white matter bundles in the brain. Okay. And besides that, these are the three things: the structure MI, function of MI, and the DTI. All these basically relate to MI machines. Okay, you're using a single MI machines, 3 different acquisition protocols to acquire the related to the images. And but there's another popular things these days is functional imaging data and modalities are called MEG or EEGs. And MEG is technically for mapping the brain activities by recording magnetic field produced by electron current in the brain by using very sensitive magnetometers. Okay, EEG is a recording of electron activities along the scalp. Okay, something like this. And the EEG measures the voluntary fluctuations resulting from ionic current flows within the neurons of the brain. Okay, MEG and EEGs is the development of these 2 technologies has been consistently grown. Okay, even though it's not that extremely popular as CT and MIs. But the EEG and MEG is becoming a very important tool. Okay, and more studies started to connect the MEG and EEG data for the subjects. Due to one, I think the good reason is because MEG and EEGs you can acquire the data very high temporal resolutions at the milliseconds. Okay, but even though they suffer from these kinds of very low spatial resolutions. Okay, localization would be a big disaster for the MEG EEGs, but even though technology keep on improving. Okay, but in generally, and the way people are using the structure MIS with complementary techniques like CT. Okay, because if you wanted to map like a bone, mammograms, the vessel, the tumors, the CT is better tools for that, right? And also the histology is because we wanted to look at the detailed information other than structure MI. But diffusion MIs, and actually besides diffusion MIs, there are people also using the tracer studies and also doing these histologies. Okay, that's also very important. Okay, if you want to look at something deep, okay, you have to use additional tools. For functional fMRI, you're basically, besides that, you can use PET. Okay, if you are looking at some more like metabolic kinds of information. And the EEG, MEEGs, there's a very high temporal resolutions in electrical activities. And also other kinds of tools like NIS, TMS, and electrodes, okay? And this really depends on your targets. But actually, in practically, and all these kinds of complementary techniques are designed to measure different physiological measures, okay? And these are the three major things. One is electrical activities, okay? That's related to excitatory, inhibitory, or some of actional potentials. And another category is there's basically metabolic responses, okay? And like glucose consumptions, oxygen consumptions, and also thermodynamic responses, okay, like a PET, like a blood flow, blood volume, blood oxygenations, okay. That's all the different tools are developed to measure different type of the physiological kinds of measures in the body, okay. And nowadays for neuroimaging applications, the golden standard these days is you have to do structure MRI, DTI, function of MRI. You know, that's the standard kinds of the setting these days. People are acquired all these for. For some studies, people also encourage you to include the EEG, MEGs if you are looking at these kinds of higher temporal resolution signals. Any questions for lecture 1?
And for the audience online, if you have any question, please post in the chat so I can unmute you so that you can ask directly. Thank you.
I think the take-home message for lecture 1 is, first things you need to know at least if you started to work on the neuroimaging data, I think the first 4 studies are super important. Okay, you need to work on if you are working on like a CT related stuff, this is a cancer TCGA, at least NLST and also PLCO has the CT data, if you're interested. And also the type of the scientific questions that we care about, if you're looking at the current top journals like Nature Science, Nature Neuroscience, they are basically the type of questions they care about it. Is these are the several major kinds of scientific questions people care about. And also they relate to the imaging devices, and you need at least have the basic information, knowledge about each of the imaging modalities. You don't need to know perfectly. Yeah, I think that the biobank data is not open data set in the sense of, I still think that they are open data set. Okay, but you have to do a lot of documents, and also you need to pay the fees for get the access to the UCA biobank data set. Okay. But I think the amount of fees you pay is, I think for me, it cost me about £10,000 for 3 years. To me, that price is very low. given the amount of effort they spend on connecting that data set. This is the largest data set in the world in any aspect. Okay, you can do tons of research on that. Okay. Yeah, I will going to share the slides afterwards, not right now. Okay, don't worry. I will post my slides, either I send an email link to you, and also I post on my website. Okay. And also for each image modality, you don't need to know everything. Okay, first you need to get to the data and start to play with the data. Okay, that's important. So not just read too many books, because each of these image model, you can think about the number of papers associated with each image model, maybe thousands or 10,000. You don't need to know all these methods and know all these methods. You just need to read a few review papers and can start to work. Any questions?
Oh, actually, Professor Zhu, I have a question myself. So I know that in the adenine database, for example, the fMRI data, there are some raw images and some pre-processed images. So like when we do the analysis, do you recommend we use the raw images and do the pre-processing ourselves, or just directly use the process by the adenine?
Yeah, I think this depends. And this depends on what question you wanted to address. Okay. And if you just wanted to work on the downstream analysis, you just say, I want to ignore the pre-processing steps. I only work on the processed data. In that case, you just, if they share it with you, you just use it. At the least, they are more experienced than you do. Okay. Yeah. And if you are working on the level question related to the imaging processing, okay, like doing segmentation, reconstruction, and ray tracing. If we want to do something related to that, you have to work on the raw data.
Okay, I see. Thank you.
This really depends on the type of questions you're working
on.
Right. I did most of the data processing by myself. Okay. But I use most of these tools from the public domains. I know which tools is good, which are not. Okay, and also we modify some of these tools.
Thank you.
Maybe we'll take a break for 5 minutes. We resume at 10, okay? Can you see my screen?
Yes.
Okay. Okay. Let's start with the second part. And about the image analysis method. Okay. Yeah, list of references you're interested in, you can take a look at them. And in these days, the deep learning method is the dominant method in the field. Okay, it represents of this state-of-the-art method in the medical image analysis. Even though the traditional method is still there, but is the majority dominant the method based on the deep learning. Okay. And this is the first part is about overview of the imaging analysis methods. And in generally, for imaging analysis, usually there are two levels of image analysis. One is related to the individual image analysis. And another one is really population level image analysis. For individual image analysis, at the least, there are three things that are critically important. Three set of type of methods. The first one is imaging reconstruction. Like you have the raw, you get this raw data, okay, and from the scanners. Even though in the scanners, they already process the data. That's not the true raw images, okay. That's already processing image the data from the components. Okay, they basically they output the images, and basically you're taking the images, and in most cases you need to do another level of the reconstruction. Okay, and this is the first things you need to know how to do it for each image modality. Okay, the second things you need to know is relate to segmentation. Okay, for image analysis, what is the number one kind of task in the image analysis? Is the segmentation. Okay, these days is very, you need to remember that's the basic of the what do you call the segmentation? We're going to talk about it. Okay, and the key ideas of segmentation, just you want to identify the objects of interest in the pictures. Okay. And another level of questions used to be very important, and it's called the registrations. Okay. And basically you're trying to register the brain or different organs to a template, okay, all called atlas, okay? And when you're doing neuroimaging data analysis, there's all type of atlas in the field. Which atlas is better? There's no conclusion on that. But some atlas is more valuable than others, okay? And when you have the atlas, basically the registration is basically pull all these the images from individual subjects and register to the template. Okay, you try to establish the coordinates or kind of thing. And besides that, your another individual level analysis is basically you're doing multimodal analysis. Basically, you integrate multiple imaging data together to get a big picture about structure and function of a particular subject, okay, for a given organ. This is basically imaging, individual image analysis. The three major things: reconstruction, segmentation, and registration, okay? And if you, for any imaging modalities, if you want to process data by yourself, you need to know how to do it, okay? And then in practice, when you have all these imaging analysis tools, and basically you can extract important information and for this detection, diagnosis, surgical planning, and prognosis, okay? And that's the first step. You need to work on it. You can use these tools to extract all these kinds of information for all these different level of questions, okay? Then you need to transfer the data to knowledge. And in generally, there's imaging processing steps. You're doing the raw images. Then you first go to reconstruction, image segmentation, and doing the registration. Sometimes you add a smoothing or not. That's not critically important. Only if you want to basically doing population statistics, then you may add these smoothing tools inside. Okay. And these days, you have these kinds of the left three panels relate to image acquisition, imaging pre-processing, or data analysis or interpretation. The underlying kinds of method that people are using these days is mainly mathematics, statistics, and machine learning. Okay, for machine learning is many of the deep learnings. Okay, all these kinds of like SVM type of. For image acquisition, you need to know what is the signal models, okay? How do you describe signals? The second thing you need to know is noise resources, okay? What kinds of noises in the different noise sources in the data, okay? That's important, okay? And after you know that, the second is the imaging processing. You need to know representation, or how to represent your images. Okay, that's also important. It's not trivial, okay? It's not just a simple and 3D or 4D data, okay? And also you know the segmentations. What kinds of objects you want to segment out? Why these objects are important for downstream analysis. The third thing is you need to do registration. You need to register your data to the atlas. How to build the atlas. What kind of analysis are useful for downstream analysis? That's all important things. The next things is related to statistics, the population level analysis. Basically, you're doing statistical modeling and inference. Okay. And for imaging processing, and mathematics is quite simple, and by notation. Okay. And the F is the images, the input data. The T is operators. You'll get F hat. F hat is the features you want to extract. That's the kind of unifying the math notations. It's easy to say, it's difficult to do in many cases. Okay. And this is most of the imaging processing problems are opposed to the inverse problem. Okay, that's the reason many people are working in these areas. And in particular, people from applied maths. They formulate everything out of math problems, okay? And that's not always the case, okay? the more I work on this kind of applied problems, I appreciate more on other aspects. And you have these kinds of you have these kinds of small f. This yellow box is your input images, your quiet, okay. And your target is this apples, this capital F, okay. And but actually in practice, you have the small f, that images you dump into your developable tools, T, capital T. You get f hat, okay. That's basically the extracted features objects, okay. This f hat may be just an orange, okay. And in many cases, the orange and the apples looks like in shape, but intrinsically they are maybe different, okay. That's the reason we keep on having more papers in the average, okay. But in generally, the distance between the f and the f hat are not exactly equal to 0, okay. Sometimes even the distance is high, okay. This is about imaging, and also for population level imaging related analysis, okay. This is nothing. When you finish the individual image analysis, okay, the f hat, when you get to the f hat, okay, you dump into the population level analysis. In many cases, you will find that it's not that critically important that much, okay. And the imaging process is important, okay, because the garbage in, garbage out. That's the reason I personally spend a lot of time work on the first level individual image analysis, because I want to control the signals extracted from imaging data, the quality of these image features, okay. That's the reason I focus on shift at least 50% of my time on doing this part, because I think that's super important, okay. And if you only work on the population level analysis, you will ignore many important things in the raw data, okay. And I strongly recommend that people should shift at least half or 30% of their time on the individual image analysis. There's a lot of imaging the problems, statistical problems associated with population level analysis. The simplest is group comparisons, group analysis, and you then quantify the longitudinal changes of the images. And also you can use imaging as the predictors to predict the clinical outcomes, okay. And also you can do correlate imaging with the genetics or other image data type, okay. That's the imaging genetics we're working. And what are the current important questions associated with the population image analysis? Yeah, this is a list of questions that I have been working on. And the most important things these days, people wanted to do the column or mediation analysis. Because when you have the data across different types, domains, and how to really link all these different data types together, both stationary and also longitudinal, okay? And that's basically something is critically important in this days. And also these days, experiment design becomes super important. And because for most, we used to have random samples. These days, we do a lot of case controls, And in practice, when you try to connect the data, you saw from these larger scale studies, always have these kinds of the sampling bias when you try to connect the data. And you have to deal with these kind of issues. And also you try to, because these days when we try to making causal inference, you need to do intervention. In particular, like you take the new drug, okay? Also, you wanted to basically decide the drug. You also kind of doing like the training, okay, like a specific training program. And basically that's the kinds of another level of intervention people use. But that's also very important. Okay. if you want to really make the causal in the kinds of inference or mediation analysis. The third question I've been working on is data, basically how to really integrate the data across different types. This is very important, okay? And we are not just integrating omics data, different omics data. You know that that's a very difficult task. And also these days we try to integrate all these different imaging modalities with the genomics data, okay? And also clinical, also environmental data set. And also the data that dimension reduction method is popular, but the current method usually just PCA, ICA plus autoencoders. Okay, there are three methods that are most popular ones. Okay, but in practice we just use these three just to do most of the work. PCA plus ICA plus the autoencoders. Okay. And this we can do pretty reasonable good job for most of the tasks. And also this base I've been actually working on the imaging genetics, and that's one thing. And also the imputation method, okay, because the missing data problems, very major problems for the imaging related to larger scale studies. Okay, there's a lot of block missing problems inside. And also there's from randomly missing all the biased kinds of the missing lot and random problems inside. Okay, that's the reason we call it imputation method. Okay, and also we are interested in like knowledge-based systems, because when there are thousands or tens of thousands of the existing literatures, how to integrate the existing findings together. That's called knowledge-based systems. That's one of the things that I've been working on. It's really using the knowledge graph and try to integrate all things together. Another thing is object-oriented data analysis, and basically that's related to you're working on your object. Could be a function, and it could be a matrix, and could it be the manifold value of the data, could it be a network data. That's the different data types. My colleague Steve Marin and also Dryden, they wrote a book on the object-oriented data analysis. You may want to read their book. They basically, there's all the different data types and generated from the imaging data. You can use that for a lot of method development. And also predictive models. And I think most of the statisticians work on that. And just, like a traditional regression analysis, survival analysis, and the multivariate regressions, and all these deep learning based method or ensemble method. That's all the different predictive models. And I think that you're familiar with. And also the statistical parametric mapping that's related to traditional population statistics in the neuroimaging field. In the traditional statistical parametric mapping, just the feeder regression models, parametric or either parametric or semi-parametric, then do the multiple comparisons. I think you still can do it, but that's not a hot topic these days. Okay, and but this because this used to be very important, but nowadays just is kind of the opportunity over there is quite limited, because the traditional method and you're doing some, it's more like you can do some simple extension of most of the existing methods. You can get these things done. I think the current days the most important topics like we're thinking about is causal and mediation inference. Experiment design is super important, and also data integrations, imaging genetics, imputation method, knowledge-based systems. There are a lot of opportunities in these directions, and for like for the dimension reductions, you can work on it. Not too much can be done, and compare with your existing methods. And also, there's a lot of object. If you have some new objects, you can, of course, you can do development. Also, and you want to do too much on it, given the existing tools are pretty powerful, and also statistical parametric mappings. Okay, these are the major topics you can think about it. And if you choose one topic or multiple topics to work on it. Okay. Any questions? Yeah, but in general, this is a ecological layout for the imaging related data analysis. And I have been giving the seminar talks on this topic of the last 2 years. The first thing is when you work on the neuroimaging data analysis, first things, you need to build these kinds of large scale database. Okay, you need, even though you didn't connect the data, you can still can sample the data from different public domains and to build this larger scale database. That's super important. Okay. The second thing is when you have all this data, you can do. You don't have, if they already process data for you, of course you save tons of time. But in generally, there's a two-level question. One is deconvolution. That's related to imaging reconstruction, and also learning. That's also related to segmentation and registrations. Okay. And also you can integrate the imaging data with other things like doing data integration or predictions. Okay. That's the basic of 5 major areas you can work on. If you really wanted to do this neuroimaging data analysis, there's 5 things you needed to think about in the first place. Okay, these are kinds of big pictures. Any questions? Yeah, I think that's a good questions. Okay, GBO asked the question regarding to may I know how much computational storage resources we need to analyze and save the UCAP bank data, or can be cloud-based? Of course, you can do it on the cloud, but you need to have money to pay for that, the storage and also the processing fees. For me, I use the UNC the resources. Majority are free. Okay, I don't need to pay anything at the UNC for that. I just need to buy some hard drivers, and also there's your university has some resources basically for process the data. If I don't touch the data, I just want to put it somewhere. If I don't touch my data often, then I just put it there. It's almost free. Okay. And for the process, if I processing some data right now, I use some disks I bought and put over there and upload it to the service and the process. After I finish that, I just download it out and put on just some disks, and so that I can save a lot of money. I think about the UNC, at least I can process this data without charging me anything. Okay. But I do agree is processing UCAP bank data takes years. Any image modalities. If you process like a functional structure MRI using FreeSurfer, like DTI stuff, if you're doing some detailed processing, that takes years. Okay, because for some of the processing, your text for one subject takes around 24 hours to 10 to 24 hours for one subject. If you think about it, you have about 40,000 subjects. How many computer hours you need? Yeah, if the university charge you for that, you could get bankrupt for doing that. That's the fact. But actually, UCAP bank did process the data, and some group then you can download their process the images. And the NESS, my collaborator Stephen Smith processed the UCAP bank data. They extract about 3,000 of imaging phenotypes. You can use that data set. They are pretty good. Okay. Yeah, I think that in any case, if you want to get into, if you're not familiar with these areas, first things you needed to go through my lecture 1 and 2, think more carefully, pick up the data set, pick up the questions you're interested, and try go through all the things. Even for me, I have been working in this area almost 20 years, okay? It takes me years, long time to build this up. It's not that easy, okay? It's not that like you're working on NASU type of problems, you have the theory and you do it. You can do by yourself. And for this kind of things, it takes years to do it. You need to learn how to do it, okay? That's the reason we need to share our experience with the community so that people can get into the field pretty fast. Yeah. That's the reason we need more people to get inside, okay, to work on this kind of really large scale data set. Okay. We need to change the statistics in a certain degree, okay? Statistics, I think this is a completely new generations. Okay, that's a completely new type of problems people are working on these days. Okay. And I will go through these kinds of the individual image analysis pretty quick. Okay, if you have any question, you can ask me or stop me or anytime. And first thing is when you have this imaging data. For MRI, usually they have the .com format, okay? And that's created by either by company or by a set of kinds of the companies of the researcher groups. They set up some standard, okay? Basically, this is the data format we decide to use, okay? And all other people can have to follow it. DICOM is a standard for handling, storing, printing, and transmitting information in medical image. Okay, usually you start with DICOM format. That's the standard format for all these big companies like GE, Siemens, or other kinds of related companies. Okay, if you wanted to, that's the companies that produce these kinds of devices. And actually, this for the medical MRI images, there's a different imaging. Usually, they don't directly work on the DICOM. Okay, that's the DICOM. Usually, you have the imaging devices, and you get to the data. That's the DICOM format. And when you're trying to process the data, usually people change it to. They add additional information in the DICOM inside, and actually create additional kinds of data type. Okay, usually that's the more researchers go into that areas analyzing the data, they find more problem associated with the original data format. They will add some new features. If the people reach agreement, they just change the way how to save the data. Okay, that's usually the format is evolving across the times. Okay. But in general, I won't go to the details when you read the data. There's a different data type. You have to be very careful when you write the code to directly import the data into your computer. Okay, there are some subtle points, subtle information in the header files. Okay, usually for imaging data, you have header files. The header files could be an independent file. Okay, that usually contains a set of important information associated with the images. Or that header file usually is put at the beginning of the imaging files. Okay, they merge together. The first 400 bits are reserved for the header files. Or at the end. Okay, and usually they reach some agreement for doing that. Okay, that's the reason they usually provide some important import kinds of files so that you can directly use that. You don't need to rewrite everything from scratch. Okay. And Nifty, another popular for the people who are doing the function fMRI, they created this kind of the format for function fMRI studies. I didn't go to these kinds of literature for a while, but in generally the DICOM and NIFT is very popular for function fMRI analysis. And this is the one thing you have to be very careful for imaging data. Usually they have header files and the imaging files. The header file contains some like important information associated with images. Okay, you have to be very careful when you try to read the header file. Another thing is in the for imaging data, in particular neuroimaging, they have orientations. Okay, that's important. And this is basically like the axial or like a horizontal. Like they cut this part, this is called horizontal, okay? And the kernel is basically they cut it in this part, this is kernel directions. And actually the circular direction is something like this, okay? This is basically the orientation, you must know it. This is the basic important information, basic and important information. And for the brain, this part is close to your eyes. Is anterior frontal, then the anterior temporal is this two part, and the superior frontal is more like something like here. Superior parietal is something like here. Then this is the posterior occipital, and then this part and the inferior temporal is the back. Okay, and this is the basic the orientation you must know when you try to read the images. And also for the medical image and neuroimaging data, and you have to be very careful with the orientations. And if you process the raw data, and many people don't realize the differences. Okay, that's super important. When you write the packages, okay, and the different packages have different conventions, and you have to be very careful. They use the RAS or LAS. Okay, this orientation is different. Okay, and for the radiological the orientation, the urine is LAS, and for the IS is well, the difference is the IS is for the neurological convention. Okay, there's basically one is radiologist and a neurologist. They use different conventions. One is LAS. Okay. And basically, this is the only difference is not a Y and a Z. Okay, Y and Z are the same. Okay, and the Y, Z are still the same. Only difference is you see that this is which one is called positive. Okay, this is more like this is a NEF-LS. This is the axis is point to this directions. And this think about everything from here is positive axis, okay? But the neurologists, they think of this part is the positive for x-axis, okay? This is basically positive plus axis left, plus axis right, okay? And you have to, and you have to be very careful if you work on the raw imaging data. Okay, and that's the convention is different. You get the data from different softwares, the convention could be different. Okay, you don't make this simple but important mistakes. Okay, because many students don't know it, then they make this type of the mistakes. And also header file is super important. Okay, and if you make some simple mistake, even the data you're reading inside is wrong. Okay, I made a lot of mistakes when I first work on this type of data. Okay, any questions? Yeah, in the image analysis, these kinds of tools are super important. And called imaging implanting, okay? And this actually is the same as the imaging interpolation, okay? And the key ideas is basically the data you're working on is discretized images, okay? And but when you try to do some operation on it, because you wanted to rotate the data, rotate the brain, you rescale it, okay, and or translation it. And when you do all these operations, the data you're working on originally is discretized. You have to, in a certain degree, to transform back to continuous images in order to do that, okay? And this is basically, this is very standard statistical problems, okay? And the imaging interpretation is essentially the same as the non-parametric regression problems. You have all these data observed in the domains. You have all the X and the Y. Okay, you observed. That's all the points. Okay, and basically, this is just non-parametric regression. Okay, this interpolation is just non-parametric function. You're either using kernel functions, kernels, or either you're using like a spline basis or basis functions. Okay, and usually the image interpretation is important. There is the manual to do it. You have all different tools to do that. When you do image analysis, this is one of the most popular tools you have to use. Okay, and because you're using all the time, because you're always doing imaging, rotation, translation, all these kinds of basic operation. And this is basically mathematical adjustment. You're doing non-parametric regression, and you have these kinds of this is the T theta is interpolation models you try to choose. I can using kernel method, reproducing kernel method. I can choose the local polynomial method, kernel method. Okay, I can using spline basis to do this stuff, or other things. Omega is basically the domains. You want to do it, and but X is basically connecting data points. You basically fit a non-parametric regression and basically catch these kinds of and this non-parametric smooth the functions in a certain degree. Okay, and then you're using that to do imaging, basic imaging operation like translation, rotation, that type of things. Okay. If we don't do it, you could introduce a substantial noise when you do it, when the data quality is not that great. And the standard interpolation method like least square, nearest neighbors interpolations, bilinear interpolations, bicubic interpolations, all these different interpolation methods you can use in practice. But when the data is not a high resolution, the different interpolation methods do give you very different results. Okay, this is the basic simple examples that tell you that this is two images. If you do interpolation, the different interpolation method will give you totally different, slightly different results. Okay, this is basically how do you do 1D, one-dimensional interpolation, and how to generalize it to the two-dimensional interpolation. Because your data is acquired, could it be kinds of regular grids, or your kind of all random grids. And when you have these kinds of data, how to really just reconstruct the images, complete continuous domain. This is another set of the kinds of basis you use. Okay. And I just give you simple examples. This original image is a very high resolution. If I do these kinds of the nearest labels using only 0.1 scales, I downsample the data, and what do I get? And then if I using bilinears and the cubic, I just modify, change this years to the bilinear to bicubic, I get something like this. Okay. There's the different interpolation and make the huge impact. And this is the give a simple examples. If you use spline interpolation 1D data, and you can get this you fit the models. The bigger BL is given. The only things you need to estimate the coefficients, and after you get that, and basically this is essentially more like linear models. Okay. And you can do the D-dimensional images using spline interpolations, and you still get this kind of the basic representation. You can also calculate derivatives along each directions, each coordinates. You can get something like that. Okay. And this is the basic. As I said, this is essentially non-parametric regression problems. Okay. And imaging people, analysis people always using that. And this is basically you observe the data equal to the regression functions plus the noise, and then you basically this L2 noise plus the penalty. The number is a tuning parameter. And your familiar statistics are always doing that. Okay, the imaging analysis people are always doing that too, because this is the traditional non-parametric regression problems. But at this days, not too many people work on this problem because not too much things can be done because it's become standard stuff. Unless you have some new settings, you have to do it completely different way, and then there's going to be issues. But in general, this is a very started field. And this is basically if you use the regional the penalties, and you can get the estimator like something like this. Okay, and this is for different regulation, like the first line with W equal to M, and also we have different regulation terms. Then you can get the different things, slightly different results. Okay. And this is just to show you that how does the tuning parameters play critical role inside. You see that this number equal to 0, number equal to 10, this number equal to 1000. Okay, these are original images. If you want to become a continuous images, you can get something like this. Okay, you do change the original images. And also in the imaging analysis field, there's another besides interpolation. And another thing is very important, imaging representation. When you have the image like 1000 to times 1000 or 200 times 200, have images, how do we represent such images, okay? And actually in the field, there's a method called the image representation. They usually take this multi-level approach. And what does that mean is basically they use a finite number of interpolants, okay, based on the data at the different scales, okay? And the idea is you have the raw data, very high resolutions. And actually your downsample the data in a different scales. That's the reason this is called a multi-scale representation. It's basically you have these kinds of discrete parameter L ranging from coarse to fine levels. Then you can get to the data at the different levels or scales. Then you can build the kinds of interpolants models for all the data. Okay, that means for each of the levels you have these kinds of representations. And then you have another level of representation. Basically, you have these things representation across all different levels so that you have the multi-level of functions together. You have multi-level of representation of single images. Okay, you can think about it at the coarse levels, the number of points is very few, but they capture some of the major information inside. It's a kind of, it's more like a wavelet decomposition or like something like that. Okay. Or Fourier transformations. This is basically something people are working on the imaging analysis. That's the reason if you work on non-parametric regression problems, this imaging people are doing the image analysis doing the same thing. And also for imaging data, and also one thing is basically also in the computer vision is a very important topic called imaging denoising or filters. Okay. And the image may be dirty because you have these kinds of dots, speckles, stands. Used to be a very important topic, okay? Because the applied math do play a critical role here, and how to remove this noise, and also then people develop different level of filters to remove the kinds of noise in the data, okay? And this type of math is used like for 2D images. They define these kinds of called the 2D point spreading functions, and certify the certain properties. Okay, this is more like a local kernel smoothing type of things, and basically, and this type of kernels usually is good, and because like Gaussian kernels. Okay, they preserve the low frequency signals. Okay, it's more like you smooth the data, you capture something major things. They suppress the high frequency because there's a lot of jumps. Okay, for these kind of things, you don't use this type of kernels. Okay, and actually, if you want to do some theory on it, that's actually you can do 2D Fourier transformations, and you will figure out what does this kernel, the 2D representation of this kernel function looks like. Okay, in the frequency domain. Okay, this is basically we call it no pass conditions. Okay, there's a whole ton of theory associated with this stuff. You can spend years just reading all the existing papers. There's more than 10,000 papers on the topic. And also the most common in the kernel function we studied in statistics is more related to these kinds of the Gaussian kernels. And if you are symmetric or not symmetric, or orientation selective or polarized, and in generally, if the kernel function is PDF, and you can define this denoising as the convolution problem. That's the reason at the beginning I'm talking about the convolution or deconvolution. Okay, that's the basically the key things behind it. And there's a way to the average filters, and basically you have these kinds of kernel functions, and you apply to your raw data, you'll get F tilde. Okay, and you can develop your own kernels based on the data, the task, and so that you can try to do deconvolution on that. Okay. And this is a simple example to illustrate there's two different kernel functions, filters. And then basically this is, you'll see that they try standardize it to be the PDF. And so that you can get, this is the raw images. If you apply to that, you get something like this. Okay. Also, they have the boundary effect. We are not going to that detail. Okay, but then you have to deal with the boundary effects. But for high resolution images, that's minor. And also, the people are also doing like the problem with average filters, and because this is very sensitive to the outliers, and the people also develop the median filters, which is to be more robust. All these things are, the people in statistics do the same, very similar things. Now, robust statistics, you can do it. It's very minor things these days because people already know this topic very well. Okay, we're still doing the similar things, but it's a very minor topics. And also, these are the media filters and these original images, corrupted images, and these are filtered images. There's two different examples in the computer vision, almost 20 years ago. Okay, how do people do with this stuff? They used to be a very hot topic in the computer vision because that's the original computer vision. There are a lot of many people from applied math working on these areas. Okay, they dump a lot of the calculus and real analysis inside. Okay, functional analysis inside. And also they're doing the high pass filters. If you have these kinds of jumps, all these kind of things, and then they wanted to preserve these jumps. This is very important in image analysis because all these kinds of the boundaries are critically important for imaging understanding. Okay, that's the reason people develop the high pass filter, essentially to calculate the derivatives, so that you can see the big jumps, so that you have to be very careful when you smooth over there. This is the typical example. The famous lady from Italy, something like that. Then if you're doing these kinds of these raw images, if you use age detection, they essentially calculate derivatives. Okay, and you'll see that this basically this kind of sharp pictures. When you do image analysis, you have to deal with this, preserve that. Okay, this is basically this kinds of filters has been in the field for a long time. They're just doing the similar things. But the imaging, and that's the reason that many people in the imaging analysis, they all think about non-parametric regression guides. Okay, think of that. Okay, but they are doing the similar things. This is a very standard statistical problem. And for imaging analysis field, the most important things is the imaging segmentation. Okay, yes, these are the most important things. Okay. And it's the number one task. What is called imaging segmentation? Just the subdivide, you divide Is about your subdivide the image data into meaningful entities like objects, regions, boundaries. Basically, you partition the images into the disjoint object sets based on regional properties. Okay, and that's very important. Okay, basically, you have the images. When you look at it, your segment tells something you're interested or very for downstream analysis. And the mathematically also there's a lot of applied math people going inside to define this mathematically. And the definition is something like this. You have this imaging is a function. Then you have this omega is the domain, okay? You wanted to define this in the imaging domain, you want to define a mapping from omega to m. M is a set of the different sets, okay? And the image segmentations is a bridge between low-level vision or imaging process and the high-level visions. And its goal is to class the given image into collection objects. Then the high-level analysis task can be performed, okay? The object detection, recognition, and the tracing, okay? That's important. Basically, the imaging segmentation is basically your segmenting image of omega the domain into the different disjoint regions, okay? And there are some, ambiguity along the boundaries, but that's not a critically important in many cases, okay? But each, yeah, this is basically the image segmentation. You segment the domain into the different disjointed part. And this is just to give you examples like L-wave segmentation for CTs. Okay, this is the segment of L-waves. And automatic segmentation of the brain, this is the like subcortical regions inside the brain, sub-regions. Well, that's very important, okay? And actually, the imaging segmentations, they use the statistical model to do it, okay? And the idea is very simple. You have to observe the images, okay, the features. And your classification images, that's your target. That's the partition related to these partitions, okay? And you can construct likelihood functions, the conditional probability of y given x. What's your X is latent, you do not observe, but that's why you observe the images. Then you can impose the prior information on the X, okay? And you can get you get the Bayesian rules, and you can get something. You try to use given observed data to make the inference on the X. And then basically this is the Bayesian rules. And this is basically you can use in posterior, mean posterior variance because you all the students from statistics. This is basically latent variable models, and basically you need to estimate from y given x and all the prior and to get to the y given x given y. Okay, this is basically the statistical models for segmentation problem. The problem in itself is not. The formulation is still correct, but this is basically, I give a simple example like Gaussian mixture models. How do you use the segment out the boundaries of CTs, okay? And that's because these have very sharp contrast of the lung, okay, and other surrounding areas. And you can easily get to the CT boundaries. And this is the original statistical model. It's still a dominant method in the field. The formulation is still correct. Another thing is the people from applied math, they're using the deformable model segmentations. The essential ideas is we tried to formulate the problem as the deformable models. That means they try to target the boundaries, okay? They think that the boundaries should be the curve or surfaces, okay? And then they try to estimate the surfaces of that, okay, the boundaries, okay? And that's the target. And they're still using these kinds of, they build these kinds of the gamma as this union of the boundaries, all these surface occurs. Then they input these kinds of the likelihood functions and the priors. Okay, they join to solve that one to get these kinds of gamma hat. But you know that this gamma is non-parametric regression problems. Okay, and that's the reason you have to use a variation method to do it. Statistics, most of statistics are not good at this because this is pure non-parametric problems, and you have to solve this optimization problems. Okay. And this is basically there's thousands or 10,000 papers on this topic, okay? Like these parametric active contours, how to model the boundaries, okay? And then you have to represent it and you get something like this, okay? And this is used to be very important topics, okay? And just to show you some ideas how to do it, okay? You can nowadays using the MATLAB for these simple problems, you can easily draw initial values. And then they can automatically to solve that. Okay, you see that? Yeah. But these days, people don't really use this that much because they simply was beaten by the neural network. Okay, and nobody really using this kind of method because this kind of method is very sensitive to noise. Okay, the performance is not great. And this is the neural network, and basically they still, you know that this is still non-parametric mapping, okay? Because this is essentially, you want to get the object function I, okay? This is non-parametric functions. Deep learning is more powerful than that. And then that's the reason the whole imaging analysis field, segmentation field is whole swiped by this. And the most standard method is just UNet, okay, these days, You have the annotated data set, you just put in the UNet and tune the structure a little bit, you will get a pretty reasonable, decent job. You don't need to really go into this applied math method at all, okay? It doesn't, these days is just simply people, in the field, people simply use UNet-based segmentation method, okay? And another topic is called the imaging retractions. And it's basically the process of transforming different data set into one coordinate system or multiple coordinate systems. In the neuroimaging analysis, all these organs, this simple coordinate system is super important. Why is it super important? Because this define the dictionaries or define the standards so that the people can compare the results across different studies and all different subjects. Okay, that's the reason the imaging retractation is super important and for the kinds of understanding and interpretations. Okay, that's very important. And for when you're working on these areas, to me, there's two level questions quite important. One, how to define these atlas. Okay, that's super important questions because all these downstream analysis in certain degree depends on it. Okay. In particular, you wanted to interpret at the individual levels. This is basically, there's something wrong with this part of your brain or this part of your heart. And whether it is, what kind of function associated with that. This kind of questions are super important. Besides that, the retractation is the self. And it's basically you need to understand how to register different brains to the same coordinate system. And you need to understand what is the retractation is doing, okay? And the retractation is not to change the original images, okay? Actually, when you have the template, the essential ideas that they try to build a mapping, one-to-one mapping between the template of atlas with the individual images, okay? So that the different subjects, they can compare over each other because they can always map back to atlas. Okay, they only change the coordinate system. They just the basic says that for my brain, this point correspond to which point in that atlas, Then when another person says that because this other point on the edge is corresponding to my point here, so that those 2 points can compare across object. Okay, this is, you'll see that the deformation or transliteration is, you define this T, the coordinates. They didn't change the imaging measures on that. They only changed the coordinate system. They basically, they provide a unified coordinate system on that. And therefore, in the image analysis retrations, you already start on 2 pairs of retration. And basically, you have 2 images on tau and R, and you find transformations between their coordinate system. Okay, and then you basically you try to the 2 images when you're using different coordinate systems, so that you can map them together. This is usually the L2 north or other north functions. Could be Wasasa distance or other distance, L2 distance. S is basically the smooth penalties. You put a penalty on this transformation field. You want this one to be relatively smooth locally. Okay? This is basically the image retration about. You don't have to involve too many subjects, only 2 pair of the images. One is a template, one is the individual subject. There's a different, this transformation function could be as simple as the kinds of parametric models. This is, you have these kinds of different scaling, translation, shielding, rotation, general affines, then like linear affine. This is all these kinds of things is the parametric model. It's just like a linear model. This have the finite dimensional representation of the transformation field. And also you can do non-parametric transformation. It's so similar to non-parametric regressions. They essentially just accumulate all these things locally and merge them together. And this is basically, you'll see that this is the transformation function is about. Okay. And just to show a simple examples of how to do translations. These original images, you're doing some shift around. You see, that's what you get. You also can make it bigger. Okay, and this is also another type of the standard translations. And this is basically all some like nonlinear translations, and this is very, this very weird translation, very severe translations like this, and this is like a rotation. Okay? And actually for the original retrations, and sometimes people are not, usually there's feature-based retrations or like the raw image-based retrations. And the feature-based retration, they basically sometimes you just choose some landmarks, okay? Like your hands, your tick points, and other points, key points in your palm. And basically there's another hand. You want to register these 2 hands together. And basically you're choosing this is like 1, 2, 3, 4, 5, 7 points as your key angle points. And basically there's a pair of the things that you match them together. Okay. You usually these 7 points represent one hand, another 7 points, another hand, you can register them together. Okay. And then you can basically doing this landmark based retrations. And then you can calculate these kinds of the L2 north, and then you can represent this as a regression problems, because they're typically just a regression problems. Because this is just a coordinate mapping, just a non-parametric regressions. Yeah, you want to find the T, okay? You want to find the T, the mapping to your 2 images, okay? To coordinate systems. That's the basic. That's why people these days just use deep learning to do it. Yeah. And then you can using reproducing Konohara space to do it. That's the original people doing that way. And you ask, could it be just the replacing kernels? Then you can just calculate that. And then you can just register the different and this landmark-based retration to do it. Okay. And then you need in practice, people, you have a different, when you have the single images, you can try different type of the features, combine them together, do iterations. Yeah, there's a lot of traditional way to do that. But it is a typical regression problem. Okay, if you are familiar with this, this is just a regression problem. Okay, but that's a regression problems at a single image level. Okay, single two pair of the images. One is a reference, another one you try to match with that. It's all go back to regression problem if you think of that. Okay. And what are challenges? You know, you've been working on this area for long time. What are the most important challenges we are facing these days? And I just share my personal opinion on that. And actually, this relates to your individual image analysis, individual level image analysis, or population level analysis. And I think the individual image analysis, most difficult problems is annotation data set. Okay, when you do all these segmentation registrations, and if we want to really doing great, you have to have a good data set with annotation information inside. Otherwise, it's difficult to justify, to validate your segmentation results. But that's super important. For me, that's the whole barriers of the whole field. Okay, and also when you try to do image reconstruction, you have to take account from statistical perspective of this. There's a lot of complex objects inside. How to make sure that your processing does not change the object. Another important thing is how to take account of the spatial temporal structures in the data. Both are correlation and also relate to the mean structures. And also imaging data analysis, and there's a lot of issues related to computational power and also computational tools, because you need to handle extremely high dimensional data, how to do the efficiently. Okay, and that's very important. Not just I develop very fancy, but it's a computation impossible to handle it, to process the data. And that's useless. Okay, in practice, you always think about why, if I develop this method, why is the computation feasible in what sense? How much you gain by using this fancy method at the cost of a lot of computing power? Okay, you need to justify that. And also for the individual levels, for the population levels, you have to you're aware that there are a lot of complex things related to like the complicated causal pathways of the different disease. You know that imaging data is just the one way to take the picture of the functional structures of the organ. That's the basically, you have to put them into the kinds of how do you really use it to map the etiology of the certain disease. And also there's tons of complex missing data pattern inside. I don't think the standard, even though we have done a lot of missing data problems, I don't think it's the kind of statistical missing data problem method are good enough to handle that. Okay, and I know that some people are working on this. Okay, I'm working on it, but I think this is very important topics. And also you have to handle heterogeneity across subjects, studies, and population. This is also important. Yeah, and also there's a high dimensional variables across the different domains. How to do it right, do it correctly. It's both we are, and there's a lot of the data limitations and other things we don't really know, but that's great topics to work on. Also the sampling bias. Okay, anytime you develop a method, you want it to generalizable to different populations. And that's a very subtle question related to sampling bias. Okay, and that's basically the major challenges I'm facing these days related to individual and the population level analysis. And that just to give you some sense about this, what's the data challenges. The most important lack of the number of the annotated data with high qualities. That's the reason I emphasize that basically in these days, if you work on any specific application area in data science, the most important questions is how to clean up your data. Okay, it's not about to do group analysis. If you have to clean the data, you can do, don't need a lot of fancy tools at all. most challenges, barriers in the field is the data. How do you get to annotate the data to truly validate all the tools you developed? Okay, that's the most important barriers needed to conquer. And also how to address the heterogeneities. If you do a mega analysis, you want to really integrate the data from different studies together to do group of kinds of the joint analysis. I think you still need to deal with the heterogeneities. I think there's some people in Japan, they do this the combat. Okay, that's the one way to handle it, but doesn't mean that that's the right way to handle this type of problem. It's a nice try. Okay, how to handle the data across different studies, different scales. It's not a very trivial problem. Okay, and these are very important topics you need to think about. Okay. And another thing is how to really integrate the information across different domains for downstream analysis. It's not very trivial at all. Okay, you need to know a lot of things. Okay, you need to have a lot of the tools in your tool set. Okay, so that you can choose the right things and integrate all the tools together to solve your problems. I strongly recommend all the students has to be, good professional coding guy and also know how to deal with the data. That's super important. And also another thing is many questions. The neuroimaging really need to go to the cause, in the sense of you think about this kind of, if you really want imaging to play critical role in both diagnosis and the prognosis, and also doing the clinical trials, you need to really understand how could the imaging markers can be used, integrated with other biomarkers. Okay, let's finish the second lecture. Let's stop it for 10 minutes, then we'll resume. Okay, any questions? Yeah, I think in this lecture too, what do we, I just go back to give a rough review. Okay, the first thing is you need to know the image analysis method. There are two major image analysis methods. One is related to individual image and data analysis. Another one is related to population level analysis. Okay, for the individual image analysis, there are three major methods: construction, recommendation, and retration. Okay, and for the population image analysis, these are what are the major methods, and these are the things are linked together from the big picture. Because for us, we are not just imaging processing scientists. We are really doing a lot of integration. We're statisticians. We're doing something big. Okay, there's a lot of tricks behind it. Any questions? No, I think the T is the balance between two. The transformation functions, you're using the data. Okay, you need to use that's the reason you see that. Like this is simplest, the two hands have registered together. This is basically they extract some key features and then basically mapping it with each other. Then you can impose the prior information, these penalty functions. Okay, you can impose it based on your understanding. Okay, you can construct this basis. Okay, there's a two way to do it. This stuff, the psi j could be the existing like a kernel basis or spline basis. The only thing you need to do to calculate the alpha, or you wanted to do it as something like I can construct a precise J, like a dictionary learning method. The essential ideas is I learn the basics based on the data. And then I put it inside to estimate the alpha. And these ideas people are already exploring. Okay. Any other questions? Okay. I hope when I teach you the first lecture one, you'll have big picture about the type of applications and where you can get the data, where you can basically, what kind of type of question people care about. Okay. For second part, at least you have basic ideas about image analysis. The key image analysis essentially is non-parametric regression problems, but it's more general than the standard regression problems we are working in statistics. Because there's a different application, the data type is slightly different. Any questions? If we don't, we resume at 11:25. Okay, let's move to the third one. Any questions? Yeah, now we may move to the lecture 3. Okay. About
excuse me, Professor. There's a question in the chat about the neural net.
what's it?
Can you see the chat or?
Yeah. Yeah, I think the neuroimaging, neural network now is a dominant field for everything. Because all these segmentation, registration, and also smoothing, and like super resolution, these problems essentially is non-parametric regression problems. And the deep net is performed pretty good compared with most of the existing method. But actually, for the tradition, if the traditional method works very well, I don't think the people will use neural net. But if you some change your problems, if the existing method doesn't work well, usually people use the neural net. But I think in any case, the neural network methods are dominant method in the field instead of other methods. Just you go to the, but it doesn't mean that the traditional method doesn't work in many standard problems. Okay, just it's not as good as the neural net in many subtle problems. Okay. Traditional method, they works on the larger scale problems, and also they're doing decent job, but it's not good enough. If some problems need more kinds of refined works, the neural network should be better than traditional. And there's a direct fine imaging to the brain atrophy. That's a good question. I think that's the different aspect. I think imaging, the genetics does play a critical role in the brain atrophy. Okay. And then there could be other things like the nutrition, or like your activities, like exercise, your lifestyles, that could confound the factors. I think brain atrophy won't be caused by a single, in many cases. And for Alzheimer's disease, this depends on whether it's early stage or early onset or later onset Alzheimer's disease. Early onset Alzheimer's disease, I think the genetics play dominant role inside. Okay, but for late onset Alzheimer's disease, genetic play a very important role inside. But also your lifestyles, the nutrition, the type of food you're eating, and also other things that play a critical role inside too. But that really depends. Oh, the other one based on my own paper. The picture I show you in the last slides, that's based on my own paper. I can share the paper with you guys. Yeah, Yeah, I think all the things I teach you in the first 2 lectures are most important. Okay. No. You at least you know where you get the data, right? And when you basically what type of problems are important. And at least you have some the big pictures about image analysis method. That's the 3 things that to take home message for the, and also what kinds of population level the image analysis method are important. Okay. That's the 3 take home messages I suppose you get from the lecture 1 and 2. Okay. Any other questions? Yeah, in that case, we'll move to the lecture 3. And actually, I just go through this MI related to the image analysis. Okay, basically, we are covering the 3 major topics. One is the structure MI, function MI, and also diffusion DTI data, and related image analysis. And we will not go to all of that detail because each of these image modalities, you know, they need a kind of 1 semester to cover all these major methods. But I teach at very high levels, so that you can go to the literature to find the references. Any questions now? The part 1 we are going to cover the magnetic resonance imaging and just give you some basic ideas inside. And this basically, you know, this work is deserve a Nobel Prize. Okay. And some of these references I just pick up from my friends or the other review papers you can take a look at. And there's many model-based imaging reconstruction methods for function for MRI. And what is the MRI machines? And this is basically the principle behind it. The subject is placed in MRI scanners. Okay, you line the inside. The nuclear of H1 atoms aligned with magnetic field. Okay, the second thing. Okay, and third things within a slice of the brain. And you know that when they acquire the data, they acquire the 2D first, and then cross all these slices, then create a 3D. Okay, for most of the imaging devices, they always cover the 2D, many 2Ds add together becomes 3D. Okay, and within a slice of the brain, a radio frequency pulse is to use the tip to align the nucleus. Okay, that's another magnetic field. Okay, and that's the radio frequency. Okay, once the pulse was removed, okay, the nucleus try to return back to their original aligned positions. Okay, and then they introduce the current in a receiver coil. Okay, there's a receiver coil. Then a signal was created. And then you receive 1 number. Okay, you remember this. Okay, these are 5 most important steps in MRI machines. You put a subject in the scanner. Okay, and because there's the MRI machines scanner is in the sheltered the rooms. There's the very strong magnetic field inside. You know, then they start from 1.5 Tesla, 1.5, 3, and these days 7, even higher Tesla scanners. Okay, for humans, most commonly they use the 3T Tesla scanners. And actually, when you line the inside, then within a slice of the brain, the radio frequency pulse to use. They add another radio frequency pulse. That's where the MI physics come in. They are align the nucleus. Okay, and then once the pulse is they are going to remove the pulse at a certain time point. Okay, they control it. And then nucleus try to come back. Then they create, then they have generated this kind of, they introduce the current. Then the coils, receiver coil will record numbers. Okay. Then they record many numbers. Okay, inside. Okay. And this is the basic in MRI machines. This is the scanners. Okay, this is the bed. Then you can lie on it. Then this is the they have two fields. Okay, the P0 is a scanner main field. Okay, that's the you see that that's the directions. This is the P0 is a scanner main field. There's another. Then they defined the coordinate system like this. This is a longitudinal axis that's parallel to the P0. And the transverse platform is XY plane. Okay. That's basically this kinds of orthogonal to P0s. Okay. And basically in practice, you know that this is not about single neurons or something like that. And the atoms. They're talking about the billions of these, even higher. Okay. The difference is between the numbers of the protons in the high energy and the low energy states, resulting in net magnetizations M. Okay, that's basically this is the coordinate system, and the B is used for magnetic field, and this is basically the key coordinate system. You can take a look at it. Okay. And in the MRI machines, there's two parameters are super important. One is called T1 parameters, and one called T2 parameters. And what does that mean? You know that the P0 is the z-axis. Okay, the XY plane is that's important because when you put the subject in the scanners, most of the atoms is parallel to the P0 planes. Okay, and basically by appropriate Tau sequence. Okay, and basically you can align most of the atoms to these kinds of the XY plane. Okay, you're using another B. There's another B magnetic field to tilt it to the XY plane. Then you can rotate in XY plane. Okay. Then you take it away. Okay. Then take that P0 away. Then all the atoms started to move back to the Z directions. Okay. You'll see that for the T1 recovery. Okay. And the T1 recovery, because when most of the majority of atoms rotate in XY plane, their component on the Z direction is almost equal to 0. Okay. And then you basically, because you remove the P, and because P0 is always there, that's the main magnetic field. The atoms started to slowly come back to the Z directions. That's the reason there's a projection on the Z direction go from 0, getting larger and larger. Okay. Here is this is the scale. This will become to 1. Okay. This is the basic called the T1 recovery. This is the basic a curve. You can, when you acquire the data, you can acquire multiple time points and can recover these curves if you want. Okay. This is the basic T1 parameters. Okay. This is at the same time the projection on XY plane. The original, because when you apply the P0, they rotate all the atoms on the XY plane. They start from 1. Then because this is when you move this P away, the P0 is already there. This is basically the part projection on the XY plane is getting smaller and smaller, but it never goes to 0, okay? But it's getting smaller and smaller. This is called the D2 decays. This is the projection on XY plane. This is the projection on the Z axis. So that this is called the T1 recovery and the T2 decays. Okay. When if you're using this one T1 to acquire the data, using that mechanism, you get something like this. You see that then this is T2s, you get something like this. Okay. And all the things are curves. You can measure the multiple points to get to the parameters associated with the T2s and T1s. Okay. And in practice, MR image is not is the absolute values measurement. It's a relative measurement because the different tissues have the different T2 and T1 parameters. Okay, that's the basic. This is the case. You have tissue A and B, and basically you have the different when they return back, the curves. It's basically, and basically the decay rate or the kind of recovery rate are different. The slope are different at the different time points. If you acquire the data, there's a contrast between the two tissues. Okay, this is the good contrast, this is the poor contrast. At a certain time points, you're using TE or TR to do it, and basically then you when you measure, then you can see the imaging contrast. That's how do you get the T1 and T2 images? Okay. Then this is basically the one of the principle behind it. Okay. Besides that, okay, in practice, when you're looking at these images, okay, in the MRI machines, they are not really, this is the true like the brain images you measure, right? This is called ruled XY, okay? Basically the root 1, 1, root 1, 2, that's basically root XY, that's the coordinates. That's the basic image intensity in that particular location. But actually, when you measure the data, they are not really measure the root XY. Okay, the things where the each time points they only measure one single point. That's the single point is the information across the whole brain. Okay, you'll see the point. But what are the most important things is this. You know, MRI machines, they do all these acquisition in the K-space, okay? And the magnetic field gradient is used to sequentially control the spatial and homogeneous magnetic field. So each measurement can be expressed like this. The Kx and Ky is the coordinates in the K-space, okay? Because the original is the 2D images, right? You want to acquire the images on the 2D. But when you do that stuff, that's the basic, your target is the root XY. Okay, that's basically, if you do these kind of things, this is the Fourier transformation of the root XY. You only measure you measure the data as the Kx, Ky, okay, by using this formula. Well, you didn't really measure a root XY. You only measure as the Kx, Ky. That's the reason you're on a 2D plan. There's a slide, the one slide of images. You measure for different Kx, Ky. You measure that. You don't measure root XY. Okay, that's MRI image is about. The MRI image is always acquired in the K space. Okay, not in the raw space. By repeatedly alter the Kx, Ky, you make the new measurements, we gain enough information to reconstruct a root XY. Okay, that's very important. Okay, and you think about sparse learning, or the whole field is come from this. You know, applied math in statistics doing variable selection, high dimensional statistics. Where does that come from? It comes from here. Okay, so basically you have these, you want to recover this is the root the XY, okay? And basically you essentially acquire the data in the K space, Kx, Ky. And after you acquire enough Kx, Ky, you can take the inverse Fourier transformation. Based on the as the Kx, Ky, you apply to here, you get a root XY. That's the image data you see from the scanners, okay? Scanner did all the things. They have all these programs, and they do all the things for you. Basically, it's the operation from SKXY to root XY. Then you can take the root XY to do the downstream analysis. Okay, in the MI scanners, how to do this one efficiently? That's basically the level question related to MI machine reconstructions. Okay, that's the type of questions we're working on it. There's a ton of people working on it, used to be. Okay, but if you want to really develop a high resolution, spatial temporal resolutions, and this will become more. This is the level questions that goes to there. Okay, you need to do everything, manipulate the acquisition, and also data integration across multiple coils and do other things. That's the reason in the MI scanner machines, the data you get, root X, Y, you get is because they do it in the kernel, in the Fourier space, spectrum space. Wherever your data, you have the real part and imageable part. Okay, the data you get from the MI scanners, essentially, if you reconstruct it, you'll see that there's this inverse Fourier transformation. This is a complex numbers. You always at each X, Y, you always have two real numbers, complex numbers, and then you're basically using that to do these kinds of get a magnitude of space. Okay, and in many cases, people only use magnitude the images EMI, but the phase information could be important, but not majority of applications are on the magnitude. That's what all the MI images about. Okay, you remember this. This is you only measure SK, XY, and then you recover root. Basically, the data you have is complex numbers. You only focus on the magnitude. Then this is basically a simple example of the macro images, and these are the phase images. And then these absolute real numbers, absolute image numbers. And then you can have the roughly things about what's going on with this. Okay. Underneath, you need to understand this aspect. And this is basically what is the KX, KY. This is the K space. And this is the you can go from the K space to image space, something like this. You're using inverse for the transformations. If you want to go from the Fourier transformations from this root, you get this K using Fourier transformation to go back. Okay, this is the data, the one-to-one correspondence between this the data in the K space and the data in the image space. And why do people work on this image K space instead of the original image space? The reason, if you really work on the image space, the number of the sampling points is much larger than left one, because you see the data in the K space is much concentrated than the right ones. That's the reason people prefer work on the K space instead of the Fourier transformation in the raw space. Okay, that's the reason why people using the K space. This is essentially using the Fourier transformation to do it. And this is basically this is in the original image space. You will have these kinds of square. That's what it does look like in K space. Okay, and this is basically another example. Is the left one is called K space. The right one is image space. And if I put a yellow dot in the K space, what does it look like? You get something like this. Okay, and if you put another red dot on it, what do you get? You get something like this. Okay, this is the basic how does if you block, only take the square in the middle and basically, or just you chop the signal in the middle, what do you get? If you only concentrate in the middle, you see that you still see the roughly the good pictures in the image space. But for the second ones, you're losing a lot of signals. This is one of the key ideas behind it. Basically, because in the K space, you can compress majority of information in a small number of the coefficients basis. Okay, and if you only concentrate on this important coefficients, the coefficients associated with this. And actually you can recover majority, you can preserve majority information in the image space. That's the basic, the key ideas behind it, okay? And then people are thinking about how to efficiently sample in case space. And you can do the uniform sampling, and you can do the spiral sampling. And basically there's a different example associated with that. And this is also associated with you, if you want to increase the spatial resolutions, the number of the points you need in the case space, and if you do the uniform sample, okay? And this is the basic, it's important to note that there's not a one-to-one relationship between imaging and the case space, okay? And each individual image space depends on all the parameters points contained in the case space. And this is the different non-Cartesian sampling mechanisms, like this is the uniform Cartesian mechanisms, and this is truncated partial undersample variable densities along Cartesian sampling mechanisms. This people are using different sampling mechanisms in order to reduce the data acquisition time. And what is the model behind it? And the model is still a kind of non-parametric regression. And is very similar to the function and linear model in certain degree. But this you can be regarded as a kind of function in the model. Your yi is observed images. That's in the in case space. And the yi, the yi is in the case space. That's your observed data, and their relationship with the I'm going to abuse the annotation a little bit. Then you have this like the raw images in the raw image space, and this is the inverse Fourier transformation, and the Fourier transformation of that, and this is the given functions, right? And this is essentially Fourier transformations. Then you have S to this plus epsilon. Then the R is the spatial coordinates. The K is the kernels, the K space trajectories, the MI power sequence. That's basically the KX, KY. F is this is basically the raw image space, the kinds of the images data in the raw space. The S is basically the Fourier transformation of this. And basically they use these kinds of kinds of basis functions in a certain degree. You project this F on it, then basically this is spatial information. This is basically equivalent to Nobel Prize. Okay, it's a very simple things in the mathematics, but they play very critical role in practice. You know that basically they transfer acquisition original MI scanners start with the raw image space. Okay. And after they use the Fourier transformations and inverse Fourier transformations, they do everything in the inverse in the case space. And actually that really push the MI scanners to the clinic and to the research. Okay, that's the really big impact. Okay, they change the whole area, medical science in a certain degree. And this is basically the problems we try to solve. The basic goal is to try to reconstruct the raw images based on the observations in the case space. And basically this is under determining the problems, and is very similar to functional linear model. Functional linear model we're working on. Okay, you can think about this functional linear model. Yeah, this is given the basis function, then this take an inner product of that. Okay. And actually, there's a tons of research area papers on this topic, okay? And actually, compression sensing, the whole area is on this, is motivated by this, Okay, then you have this Y is observed data in the K-space. The A is these kinds of design metrics, and the F your target is the raw data in the imaging space. And the A is depends on what kind of kernel function you can construct for the design metrics. Usually this is highly related to these E's. Okay. And then you can basically try to solve using some loss functions plus some penalty. And then your basically R is to control noise by penalizing reference. And these whole areas, you know, like David Donahoe, all these people work on this problems. That's the reason there's many people from applied math work on it. And many people from image analysis processing people work on it. Many because of this. Okay. This is the largest application for the compression sensor. And this is just to give you some example of the quadratic parallelizations and how to do it. And this is basically give you some ideas about these two images. And this the true image data. And then you add different kinds of the parent regularization terms. This quadratic edge preserving penalties, you can get something like this. Okay. And this is basically quadratic versus non-quadratic parallelizations, like using some robust regularization to things. These things, you know, the applied math people already started for a while. Okay. This, you know, since the development of the MI related research method, and the whole this majority of many of the applied math people are working on these problems, and then people from the ECE also work on it. Okay, and this is basically the three. The one left one is ground truth phantom. This quadratic penalty, the Hongtu penalties. I just give you this is essentially you think about it in statistics. In last 20 years, people will work on the high dimensional statistics. All these original model come from this. Okay. The part two is basically structural MI analysis. The structural MI analysis, I just give you some basic ideas. And I think for this topic, there's you can take a look at some of these good books and just references to start it. Okay. And the key things, when you have these structure MI images, in particular T1 images, it's pretty high resolutions. Okay. And one thing is, what can you do about it? Okay. And actually, these areas have been more than 30, 40 years. Things the MI pictures getting popular, people started work on this. They always think about how do you try to use for information to delineate the growth of the brain, okay, from the structure perspective. And this is basically the free surface processing stream overviews. If you are interested, the free surface is one of the dominant the imaging processing pipelines people used, okay? And I don't know how many hours you use to process one single images. Maybe, I think usually it's about 21 day or maybe less, but in general, 10 hours at least. And you have this T1 weighted images input. Then the first doing skull stripping, just remove the skulls and also CSF, okay? And also do volumetric labeling. You see that they try to label this white matter, gray matter, all these things. And also do intensity normalization because of the different subject they have the large variation inside. They do intensity the normalization to standardize it. Then they pull out the white matters, segmented out. And then you're doing a lot of things like a surface atlas illustrations, like a surface extractions. They extract the gray matter, white matters. They get the surfaces. Then also they do gyro labeling. Okay. This is one of these the major tools people used And you can think about it, if you process using this pipeline to process, you can biobank your data. You need at least 1 year to do it. But I think they already processed, and you can biobank. You may want to download it by yourself. For you for free server, we direct download. We don't use we didn't do it ourselves. And when you have this structure in mind, what you can do about it? You can just use the scan the same subject across times and see how does the brain changes, okay, across times, the same subject, okay. And when you do the segmentations, and the most important things, you want to segment out to white matter and gray matters. And also you are looking at just some cortical subregions and the subcortical regions inside the brain. Okay, you want to do segmentation of that. And even that problem is not trivial at all, okay, because there's no ground truth in many cases. We don't have the good annotated data set, okay, in large scales. And that's one of the bottlenecks for the field. And you can take a look at the brain volumes. That's what is the science, the Nature paper is about. You're just looking at the total brain volumes, the gray matter, cortical gray, and the white matters across from the preterm to the 90 years old. That's the publications in Nature last month. Okay, it's tons of work to do that. Okay. Now, this, if I have the data, I can do all the same thing. But data analysis is not that difficult. Okay. But the most effort was devoted to assemble all the data sets. And this is basically the segmentation is not trivial at all. And because if you really wanted to do very good job inside, it's not that trivial problem. You can use the existing tool to do it. And this is the basic tissue segmentations. And you see that there's a lot of hole inside, okay? And in many cases, you need to fill up the holes inside. Otherwise, you create a lot of the problems downstream. And also, you see that surface extractions, because when you extract it, at the top is the white matter, the gray matters, okay? And that's the basic, this is called outer cortical surface. And also you have inside, the blue one is the inner cortical surface. That's the next to the white matters. Okay. And when you have these kinds of the structures in the 3D space, and then you can use a lot of the ideas from computer vision, like using these kinds of matching cube to represent these 3D structures in the 3D space. Okay, both outside and inside. You're just, there's MATLAB has no package to do it. Just dump inside, they will automatically do it. But that's not trivial. And how to do it correctly, that doesn't mean that the existing matching cube algorithm is good enough. There's a lot of downstream noise inside. Okay. Then you can create these kinds of 3D graph structures with 1 million of the nodes, and then you have all the coordinates, triangle coordinates in your data. That's the basic representation for the cortical surface. Okay, that's the data you really will work on before we do all things downstream. And the surfaces usually called white and the pale. Pale is usually is cortical. The cortical surface is basically related to gray matters. The white is just related to white matters. Basically, that's the kinds of the white and the pale. There's the two surfaces less to each other. Okay, there's one cover another one. And then you know how to segment the images. Then you can quantify the 4D longitudinal cortical surface extractions. Then you can extract the pale and the white, and this is the white, this is the pale. But then you can see how that's the growth changes across times. And also when you have this stuff, you can looking at the cortical thickness. Basically, it's this is the pole surface, is a wider surface. You calculate shorter distance between white and the pole surfaces. Okay, and that's the measurements people care about. Then you see that you have this coordinate system. That's the pharmacometric view, and also at each point you can calculate the cortical thickness. That's one of the type of the representation metrics we extracted. And then you can map the inference cortical thickness development across times. And this is the baby connection project. And at the UNC, there's a brain development in the 16 months. And then you'll see how there's one is a T1 weighted images, the T2 weighted images. They quite both because they wanted to borrow information from both and see how does the growth of that. And besides that, there's another very high level things is related to the gyrus. Okay, you know that in the brain, what is distinguishes the human brain of all other mammal brain? Okay, and they think the scientists think that's mainly because the complexity of the gyrus. Okay. And then this complexity of gyrus distinguish us the humans from the other mammals. And how to do this gyro segmentation correctly, how to extract reliable or reproducible features from that is not a trivial problem at all. Okay? And my friends have worked on this for almost 20 years. I tried to get that pushed forward. It's a very hard topic, very difficult to do because of limitation of the data limitations and other things. It's tons of work. Okay. But if you really get something reliable from that, that will be regarded as a breakthrough in the field. This is a very difficult problem to solve. But some people dare to solve it. Okay, that's important. Okay, we need to encourage people to do that. Hard problems. This really make a huge impact in the field. And then you can quantify the folding patterns. Okay, you can calculate the folding patterns. Okay, when you have this, you can calculate the height and the depth of the encodes the folding patterns. Then actually, when you based on each vortex, that's the basic from margin cube, you can calculate these kinds of features attached to each vortex. Okay, that's the type of data we have. Right, many people they don't really understand what's going on with this. Okay, that's the type of data we have, and you can map the cortical local gyrification in infants. Okay. And also, besides that, this is basically the major representation on the cortical surface. Another thing that people also work on is the subcortical regions. Okay, subcortical regions like the caudate, the thalamus, brainstems, hippocampus, medulla, putamen, pallidums, accumbens, caudates. All these subcortical regions are very important for brain functions. Okay, that's the reason. Like a hippocampus, okay, is when your brain start to have the atrophy, usually they start from hippocampus. Okay, that's the reason these subcortical regions also important. Okay, people do like a shape analysis of that, and you can do these 2D or 3D representations for the shape, and then you combine all these shapes together. Okay, but in general, for these subcortical regions, the shape is relatively simple compared with the cortical surface. Cortical surface is much harder. Okay, because given the complexity and the detailed structure inside, but the people still did it to this kind of subcortical region. Like this is the ventricles. You can quantify the shape and growth of that. Okay. Any questions so far for this type of data? Yeah, if you don't, I will go to the function fMRI analysis. Okay. Yeah, I think function fMRI has been the field for almost 30 years. And you can take a look at some of the paper. I didn't really update the references that much. I think the second one is a pretty good ones. Yeah. Then there's some other papers from the Freestone and the other groups. Okay. I didn't update the reference. And the function fMRI is a non-invasive technique for studying the brain function or activity. Nowadays, the function fMRI is the dominant tools for functional imaging, not just because there's a balance between spatial and temporal resolutions, ease of access, and the collaborative of the communities. You know, function fMRI areas have been growing pretty quickly in the last 30 years. Okay, and what kinds of data we have? Basically, during the course of your MRI function fMRI experiment, a series of brain scans are required where subject perform a set of tasks. Okay, and also at the rest. But then in general, you can do the resting function fMRI or the task function. But then for task function fMRI, it's important in the sense of your direct link the human behavior with the imaging data. Okay, basically the people give you, it's more like intervention because you really give instruction to the subject, you'll see how does the functional signals change. It's more like an intervention. Okay, they really instruct you to do something so that you see how does the imaging changes due to such a task. This is more like an intervention. That's important. Other imaging modalities at the NESS is not directly related to human behavior. Okay, like this. And also change and measure the signals between individual images are used to make inference regarding task-related activations in the brain. Okay, that's basically the function will really push link this human behavior intervention and MRI images data together. Okay, that's the reason this become popular. And when you're looking at the data used by basically for each subject, you get this time series data. Okay, usually people think about 4D. And when you're looking at the one function of my studies, you have subject levels, and each subject you could have multiple sections because sometimes you are not do single experiment. You do multiple different experiments inside. And also you have the different runs. Like one experiment, you run 120 volumes, like time the 120 time TR. And also you have then how many volumes and how many slices. That depends on spatial resolution. One is temporal resolution, one is spatial resolutions on that. And this is basically the typical function from the data structures, something like this. And when you acquire the function from studies, and usually you acquire the T1 and the T2 star images. The function images usually is T2 star images. Okay, you want to acquire. And the T1 images is high resolution. That's the text. You try to distinguish different tissues, but it takes much longer to acquire. But these days, they already speed up, but still it takes several minutes. Okay, and the T2 star is the function images, is no special resolutions. Okay, and this is basically is most of these spin echo images about. And this they also, and because you have the behavioral data, you can record the experiment designs. Okay. And this is basically the basic terminologies in the MI analysis. Basically, you need to take a look at field of view, slice thickness, and other things. And you have to be very careful. And usually the people, the one critical thing is for you need to make sure that the data align the same atlas, the reference. Here is usually people, when they take the scans, imagers, usually they choose the ACPC lines as the kind of reference, then you can acquire the data. And that's the basically some of the key kinds of parameters when looking at the data, like field of view, metrics of size, implant resolution, slice thickness, all the things, voxel size, all the things information. You're supposed to be able to get it from the header files. Okay. And the bold the function fMRI. Okay. The most common approach towards the function might just use bold contrast. Okay. And that's the kinds of the tracers, the function fMRI target is a blood oxygenation level dependence. And actually the bold function allow us to measure the ratio of the oxygenated to deoxygenated hemoglobin in the blood. Okay, that's their targets. That's the kinds of tracers they are targeting. And it's important to note that bold function does not measure neuronal activities directly, but they don't do that. They measure the metabolic demands, just the oxygen consumption active with the neurons. Okay. The historically, the basic hemoglobin exists in two different states, which depends on whether it's oxygen molecules. Each state has a different molecular magnetic properties and produce different local magnetic fields. When you have this with oxygen, that's zero magnetic moments. Without the oxygen, that's the sizable magnetic moments. They why call the oxygenating hemoglobin is deoxygenated hemoglobin. And actually the deoxygenated hemoglobin has effect suppress MI signals as such. Then you have the, you see the signals increase. Okay, you're looking for that. Okay. And this is the basic model of the relationship between the bold function MI signals and the neuronal activities. That's the basic. That's the second references I cited. You see that this is the middle part. This is the blood flow oxygenation. Okay, on the top you're targeted to map sensory, motor, and cognitive process. Okay? That's something related to your human behavior. Okay, your actions. And you are not directly measuring it. You are measuring this blood flow oxygenation. And actually this stuff you can measure by using MI signals. Okay? These kinds of relationship behind it, the logic behind it. You need to understand this aspect. And this is the create these models, the two fundamental models to model the bold function for my response and underline the homodynamic response functions. Okay. And there are two popular models, and one is called the balloon models. And usually people do the animal studies, usually they start with the balloon models. And they try to model the relationship on the blood volume, blood inflow, the deoxygenation, and also the flow induced the signals. And then try to characterize how does this component induce the changes to the bold signal responses. But this is basically for the people who are doing animal studies or develop tools for function. But actually, in real modeling aspect, people many using linear time invariant systems. Okay, they assume that neuronal connectivity constitute the input, impulse, and the HRA functions. Okay, this is the basic. These are the key things behind it for most of the modeling. Okay. Then this is a linear time invariant systems. And this is pretty easy. They just write a convolution between the stimulus functions and the hemodynamic response functions. you know the stimulus functions by design, and you roughly have some shape for the hemodynamic functions. You observe the signals can be regarded as a convolution of these two together. Okay, that's the rewrite of the convolution here. And this is the ideally HRF, the shape of the HRF. You have the rise, peak, fall, and undershoot, and this is related to second. Okay, and this is the basically sometimes you have the initial dip, and you not always see it. And also in practice, the V, this stimulus function is used by doing design. The early stage of people doing block designs, and these days the people are mainly doing the event-related designs on that. Okay, and linear environmental systems is specified by stimulus function experiment plus the hemodynamic response functions, and you see the stimulus induce the neuronal response and induce the hemodynamic and functions. Combined with that, you get you can detect by MS scanners. You get a function from my responsive polynômes. And that's the basic the data you have. This is the block designs, and the HI functions. You can get to the predicted response functions. And then you have event-related designs. These HI for functions. You get the predicted response functions. And this is the basically this is the one of the key ideas when you try to model function from my signals. Okay, you have the stimulus functions, the HI functions. You come out together, and you get to the observed the function of my signals. Okay, and this is the basically the some of the key ideas behind it. In generally, doing the image analysis, you have the function from my time series. You also have T1 images. You have to do the following three things. One is motion correction. Okay, that's the largest noise in the function of data. Okay, many people ask, what is the highest dominant noise in the function data? Just motion. Because when you lie down in the scanners, you are unconsciously move your head. Even you have some device trying to fix your brain. And that doesn't work. Okay, you always, the longer you stay in the scanners, your brain started, you're getting impatient, and then you're more started unconsciously move your brain. Even you have some device hold it tight to a certain degree, but you still try to do it. The longer you stay there, you're getting more noise in the data. Okay, and then you're doing special normalization. Basically, you need to try to map the data on the common space. Then you're doing smoothing. Any questions? And after you do that, then you have the process, the MI function from my images, then do the design matrices, GLM, and do statistical parametric mapping on this. And this is the typical pipelines and people usually do. You have T1 images and also function from my time series data. And the key things is basically the co-register to the template, and you're using the T1s to do the concentrations. Okay, basically you're using T1 image co-register your functional images within for each subject. Okay, just your own T1s with your own function from the images. Okay. Then after doing the retracement, then you put all the T1s and also function related together. Okay, you already align them in a certain degree. Then for first row, you're doing this warping to agenous template. And then usually if you choose the T1, if you acquire the T1 images, you're usually using T1 images to get this warping parameters, retration. Okay, there's a template for the T1. That's address. Okay, they register your individual subject T1 to that template. And also you register the individual function format to this functional, to its T1. When you get to the retration from T1 to the T1 address, then you'll get using that mapping applied to all these the subjects, the functional images. Okay. And for functional time series data, you're doing denoising, slice timing, and realignment. Okay. Then finally doing smoothing. Okay. Then you apply to all the functions. That's basically the key pipelines behind it. Okay. Any questions? Yeah, this is basically some of the key steps. You can do it in the FSL or the SPM, and or FNI or other things. But in general, these are key steps: the slide time corrections, the realignment, correlations, normalizations, segmentations, and the smoothing. Okay, and the key ideas is basically you're putting all the data on the same coordinate system. Okay, you try to remove a lot of the dominant the motion noise. Why should we do slice timing corrections? The reason is usually people do it because all the MI images are acquired by slides, okay, 2Ds. And the order of slides can quite a different time. You need to align the time across all those slices. Otherwise, the 3D data are quite at different times. Okay, and they are not. These are the basic given examples. The different times, the slides of the images are quite a different time. And this is basically tell you how to really align these kinds of different slides together using interpolations. And the most important things in the functional processing is to try to minimize the motion. And the minimizing the motion is one of the most important factors for ensuring the good data quality. If you have a very relatively large motion in the functional data, usually people just throw that data away. Okay, it's not a trivial to do it. Okay, and the effect of the head motions could be very major, and in particular on the boundaries of this. And you can see that there's a lot of artifacts inside if we do it. This is the typical example of the ringing effect in the functional data analysis. And the motion is substantial. That's not just the noise in the functional data. For most of the functional data analysis, you will see there's always include the motion parameters as confounding variables inside. If we don't correct it, and correct without correcting that, that's make huge difference. And this is basically when you're doing this alignment with the T1 images or the function from my data, and actually you're able to get these motion parameters like translation, rotation, these things, and you can using these kinds of as the motion parameters in the downstream analysis. Okay, and actually you can easily create this phantom simulate the data. You're doing like you can artificially to ask a subject. You can ask the subject to move in a certain degree and see how does the imaging process pipelines could be influenced by such a motion. Okay, it's not a trivial problems. Okay. And of course you can do something serious, but that's not allowed to use this type of equipment in practice. Yeah, and also there's two options for functional fMRI normalization or retraction. One is basically what I said is basically using your quite both T1 from EPI images, the functional fMRI images. You're using structured images to register T1 template at NESS. Then you apply to all these the warping parameter to all the functions. Another one is basically you create an EPI template. You're only using the function fMRI the data to do the iterations. But because the function of data is typically noisy, and resolution is not great. That's the reason the normalization is not great. But sometimes some people don't care about that much, but we still can do it. And this is basically the normalization structure to T1's template. Then this is the individual T1 images registered to the T1 template. And then after you get these parameters, and then you can apply it to individual T1s, and also apply it to the parameter to the function of my signals. Okay, apply this normalization parameter to the co-registered function of my images. Okay, this is basically one of the key steps in the function of pre-process. You need to understand that. Okay. Any questions? No, I think the function of MI is T2 star images. T1, you know, usually the structure is usually T1 high resolution star. No, they all acquired by using MI machines, just using the different mechanisms. For structure function of MI, it takes much longer because there's a high resolution. The function of MI, because you care about the temporal, you mainly care about temporal resolutions, and that's the reason you have to sacrifice the spatial resolution a little bit in order to get the results fast. But I think originally people started from 4 seconds for one volume, and these days they can push to 0.45 second for one volumes, something like that. Yeah, this is a function of MI is T2 star image. Yeah, if you wanted to read the paper, my friend Martin Linguist wrote a very nice paper. This number 4, if you want to read that, yeah. Yeah, this is some reference you're interested in. And for functional MI statistics, yeah, and this is the big pictures. But I think these areas people already work on for a little while. I personally don't do that much these days, and many just because I don't feel there's too much left for me to do. But also very crowded. I don't do this stuff. And then you do we finish the first part of imaging analysis. Then we do the statistical part. And then in general, this statistical part, usually there's 3 number questions of statistical parametric mapping. That's the usually implemented in SPM or FSL or these things. Pretty simple stuff. And also people do a lot of research on these brain network analysis and also do predictions using deep learning or other predictive models to do it. That's not my emphasis to do that. I just tell you the most important things for statistical parametric mapping. The key things is how to set up a design metrics. Okay, that's the most difficult. And in general, for function for my usually 3 level analysis, people usually do. One is the statistical parametric mapping. That's to try to localize the brain areas activated by task. The second one is the brain network analysis. Is to determine the network corresponding to brain functions. Okay, how does the different network interact with each other? Okay. And also, then you wanted to using the function of MI to predict like intelligence or like a psychological or disease status or others. Okay, you predict whether you make a line or not. You know, sometimes the people doing that, yes, make it fun. And the most important thing is, you know, that this is general linear model, just a linear model. It's multivariate linear model things. And the most important thing is it's not about linear model theory. I think that most people here a statistical background, you know, you don't need to really teach that. But the most difficult part is the design measure. You really need to understand it. Okay, that's the most important part. When you've been doing, even doing GWAS or other topics, you need to understand the data is more important. Why should I include that specific terms inside? And how to set up design metrics is very important. And the design metrics is generally is there are three major things inside. One is effects of interest. Okay, that's the kinds of associated with your design itself. And also some confounding factors. That confounding factor could influence your function of my response. And in particular, like the motion parameters. Okay, the motion parameters is the most important confounding factors. And also whether you drink the coffee or not. It could be important for function of studies. And these are all other residuals, okay? And these are very important. Then you need to specify effects, okay? And then you estimate the effects of error for the data. That's the all statistics about linear model, a multivariate linear model about. And that's simple for most people here. We don't need to go to details. Then you can do the calculate p-values and do multiple corrections. And these days, people don't really care about this topic very much because we don't think that that's too. Because the existing method works either very well and nothing important in that. I personally don't do it because I don't think that field is important at this moment. Because you know, my friend Tom also, you know, Keith, they did most of the theory behind it. I don't want to compete with my friend. Yeah, I think there's so many important questions to address. And in practice, when each of these voxels, you have these kinds of the number of scans. This is y for each point, for each vortex. Voxels, you have these time series. Okay, then the number of the effects in models, you have this x. Okay, like something like this. And this is just two examples. One is the intercept. Another one is they have the different effects. Okay, they have two kind of condition, 1 and 0, something like that. Then you have the models and you get to the result. Okay, there's a noise. Okay, and how to set up a design matrix is super important. The design matrix contains the two set of parameters. One is nuisance correlates, one is task-related correlates. Okay. And both are very important because one is related to your design. Why should I do this experiment? Another one is related to the key kinds of the confounding factors you have to control. Okay. And you need to include the low frequency noise and its many motions, artifacts related to hand motions, and the cardiac power monetary induced the brain motions movement. In my heartbeat, okay, your brain, your mood, in consciousness, I do something like this. Some people always do that. The neural response shape may not have been known, okay? And the hemodynamic response will vary in shape across the time and also across the brains. And this is the two key things. And anything if you cannot, you don't know it, just turn to the errors, okay? And this is basically one that's the key factors is how to model the motions, okay? And this is the one is related to low frequency noise, like drift. Okay, you see the drift in the data. How to remove that? Okay, the different packages use different tools. And also the physiological artifacts, the high frequency noise. Okay, and how to handle it. You really just use a temporal correlation, autocorrelation to handle it. And also like head motions, you put the regressors in the models as regressors and they do it. You put the three translation rotation into the model as a correlates and correct it. Okay. These are very important, and this is how to handle this low frequency drift. Okay, you think about it when you analyze the time series data. How do you handle it? You feed it like a polynomial, the kinds of low frequency kinds of basis function. Then you can get a coefficient and remove it, right? And this low frequency drift, you can do it in frequency domain. Yeah, you truncate some of this noise. Okay, corresponding low frequency part. And this then you have these high pass filters, and this you see this low frequency drift, and you can using that you can get something like this. And in practice you can do this low pass filters and high pass filters, and try to remove some of the noise. Okay. And then you can add these high pass filters as part of the corrects in the model. They help you remove that. Okay, if you don't want to pre-process data, you can just add that kinds of basis functions as a covariance in the model. They will automatically help to remove that. And then you have the raw data, and you do these high pass filters and the scaled. You get this adjusted data, and then you feature the model, and that's much uniform distributed. Okay. And this one examples with the motion parameters, and these have this left one is the different motion parameters, and if you correct it, this motion parameter, you'll see the data is the signals associated with the experiments is something like that. But if you don't correct that noise, you will see something like this. Okay, that means that's a huge impact, because that's dominated by these motions. Yeah, that's in practice. You need to have these columns associated with the different conditions, A, B, C, D. Okay, and also you need to specify HI functions, the basis functions. When you run those SPM type of the tools, you need to specify basis functions. Then you convolve it with this, you get the design matrices associated with all these different stimulus. And then you have these columns associated with the different experiments. Okay, that's the kinds, these parameters associated with these ABCDs are the parameter of interest associated with the designs. When you do this stuff, and actually you assume this HIF is correct, you assume that this L linear time invariant to the system is correct. That's basically for most of the functional analysis about. And I do have some friends basically tried to and do the non-parametric, like, because this essentially are function linear model problems, and also this is basically sometimes you can estimate the HI functions, then you can get this summary statistics associated with HI functions. Some people just direct model HI function, try to get this parameter, try to make inference on that. And this is general the convolution models associated with this impulse HI functions, the expected both signals. Okay, and this is very important. Basically, the expected both signals response equal to input function combined with impulse response from HI functions. And because the input function is controlled by the experiment, then for the HI functions, then there's a different way of doing that. Yeah, this is the basically give simple examples. And how does this is the red curves is the F, and then you combine with the HI functions, something like this, then you can get something like this, the green ones, and also you add some random noise inside, and you can get the blue curves. Okay, that's the ideal case. In practice, the raw data is nothing like this. Yeah. Then you can combine the HI functions to this box design. There's also stimulus functions. You get something like this. And this is basically the unconvolved the residuals. And if you convolve with the HI function to cover this peak, and you get a better residuals. Let's just give you an artificial example. Real data is going to be like this. And this is basically the block designs and the event-related design HI functions. You get predicted responses, and this is part of your design matrix. Okay, your design matrix is basically have this kind of shape inside. That's columns association. Besides the usage correlates, this is basically the set of parameters you're interested. And HI functions as any kinds of work was done around 2000, 20 years ago, and actually, or 15 years ago. And there's a different shape people are doing. This is a parametric HI functions. And when you try to really estimate this, because all these functions are nonlinear functions, the estimation is terrible. It's not easy. And the people also doing these non-parametric response functions and using some basis function to do it. And yeah, it's more faster, but it's very sensitive to the noise. Yeah, this is the HI function, non-parametric HI function, you can estimate it. And you know, I spent 1 year on doing this. I give up. I don't want to do it because the data is too much. You want to get too much improvement for doing this. And this is the different way of the basic functions you can use and to estimate try to feature the data. Okay, well, you see that the first rule is simple. Tell you if you use a simple HI. These parametric models to convolve these things, you get the actual data and the featured models. This is the substantial bias inside. If you add more flexibility inside, this is the basic just to tell you that the original, this the parametric HIF, they also calculate derivative with respect because the HI function is the function of the time and also dispersion. They calculate first of all the derivative with the time and the dispersion, add a 3 function together. That's the reason you have 3 columns. Then you fit the models, you get a much better fit compared with the first one. The third one is basically all these non-parametric, pure non-parametric. Okay, it's indicator functions. It's something like a design matrix like this. You get a perfect fit. Then so what? You know, it doesn't mean anything. Okay, it just give you the perfect fit. It doesn't. Biologically, it doesn't improve a lot of interpretation at all. And but when you set up the design metric X, then you just feed it. Then you assume this certain degree of this time series because you have residuals. You assume a time series model for that. Usually, that model also contains some parameters you can have estimated. Then you can create these kinds of. And sometimes people, the way they are doing is pre-writing the models, but remove the temporal correlations, and then you estimate it by using this. This is how to implement in the code. And then you have these things you can estimate the p-values and also testing statistics, and then you can do the comparisons across the different conditions, and then. We stop here for 45 minutes. Okay? You can have your lunch. Any questions? Yeah, we start from 1:30, 1:35. Okay? We come back at 1:35.
Thank you.
Can you see my screen?
Yes.
Okay. Let's restart again. I think we just finished how to set up the design metrics for single individual subject linear model, general linear model. And actually for the population or general linear model, and actually what the people used to do, there's a two way to do it. One is doing meta-analysis, mega-analysis. Basically, you put all the data across subjects and do these kinds of gigantic linear regression model to do it. And but actually in the field, people usually don't do that. And there's two reasons. One is because these days the size of the number of subjects getting larger and larger. Is almost possible to read all the data in the reading insight. A second thing is because the variation across subjects is also huge and is pretty large. That's the reason that it's not good to really try to run this mega analysis for medical image analysis. The typical people are doing these days is essentially just doing hierarchical modeling. They do that at one level and they basically push it to the next level and try to integrate things. And basically they decompose a larger the task, the complex big task into multi-layers and run in, and using hierarchical way to approach to do it. Okay, and this has become common in practice these days. And this is there's very well-known kinds of multi-level modeling, and it's not just a modeling the cell. And actually people are doing this stuff, and they try to do these kinds of estimation by this hierarchical way of doing that. It has become a golden standard these days, but mainly because the two reasons I emphasize. One is because the sample size is getting larger and also the variation across the subjects is pretty large. Okay. Then all other things is basically become pretty standard, and you run this multi-level GLMs, and then you try to do it. Okay, it's pretty standard these days for this big data era. In this big data era, and in many data sets, we cannot simply do all things together. A subsample or group, then grouping the results across each subset, and across all the subsets a popular method for doing this. And then this is in the neuroimaging field, in particular function of MI, people call this summary statistical approach even earlier than all these people, the summary statistical people are doing that, the GWAS, how to integrate the result from different studies and doing that. But we are doing at a subject level on this. And this is the different way to do the summary statistics, and by combine the p-values and also try to combine estimators. You can do these fixed effect or the random effect models for the type of models you're working on. And this is basically one typical example. So you're doing fixed effect models and also random effect models, how to do using empirical Bayesian method estimators for that. Okay. And this is a simple example to illustrate how to do it across the 12 controls and 11 blind subjects. And this is the auditory presentations of words spoken backwards. And actually you want to compare the two groups, what's the differences between the two groups. And this is basically the first level. You see the controls and blind people for the one specific kind of the task. And then you got second levels, you can combine the estimators together. But in practice, when you do these estimators, the beta images, usually they work on the raw beta images when they try to do these things, okay? And this is basically and how to do it. Some people are using tensor just to do it, but that may not be the right way of handling that. And this is basically the second example is basically this is the 4 different tasks of different words. And there's 12 kind of control subjects. And then the question is which regions are affected by semantic content of the words, okay? And this is basically the first level is there's the 4 words, you see that? And the motion, sound, visual, and actions. And then the second level, so you can combine all these estimates together and to make an inference. Yeah, this is basically the regions identified. Any questions regarding to the function from my data analysis? And another thing is about the brain connectivity analysis. Because this is also a big field and the topics are huge. And many people are these days working on the brain connectivity analysis across different levels. And also basically I just give you some brief ideas about how to do it. And one thing is basically the data. You have the raw time series data across all the voxels. And also you have all the lot of the covariates of interest and like age, gender, diagnosis, and also other kinds of motion parameters. And how to do these kinds of connectivity analysis. And this is a huge topic associated with that. When you, in particularly, for resting functional fMRIs, and this became popular in the last 10, 15 years. And they try to use basically they put a subject in the scanner without doing any experiment, okay? And how to process the data. The first one, you're relying on each 10 points of image to reference time, and then reduce the effect of subject motion over the duration of resting function acquisitions. And you correct the data for MI spatial distortions and remove the non-brain parts of the images, and estimate the alignment transformation between resting function and the same subject T1 and between the T1 and the population average images. Okay. And there's many different ways to pre-process the resting function from data. That even different pipelines, protocols are different. The results could be pretty noisy. And actually, yeah, there's the optional in the sense of some group agree with that, some group may not agree with that. And that's the reason there's some kinds of controversial arguments behind it. And this is basically the pre-processing steps is to try to remove the motion corrections and the other kinds of parameters, user parameters associated with that. And there are several key things here. One is the regression of the confounding time series of the data. How to do it? One thing the common approach is they take an average white matter or ventricle time series, because they suppose that the brain, the functional signals mainly come from the gray matter. And that's what the people are doing is they take these kinds of white matter and the ventricles, they calculate the kinds of average curves or some other summary statistics from this each subject and partial that out. Then basically you're usually just doing a regression and get that part out. They think that should be your standard. And also the motion parameters, and basically they also regress that out, and also remove all the corrupted time points. The data-driven structure, the noise removals, one thing they can do is using ICA and with automatic component classifications. Because if you run the ICAs, how to run the ICA properly itself is an intriguing questions. And for many cases, what the people are doing is basically looking at some major ICA component. You think that some of those components are not useful is lack of, if they are lack of the biological or neurological interpretations, they just manually to remove that. And also they also fit out the highest temporal kinds of frequencies. That's usually some of the key steps when they process the resting and function data. Okay, and after you process the data, okay, and this next level question is nanowall constructions, and that actually they generate, there's a scheme related to you're working on either voxel-wise time series or RI-based or time series to generate correlation matrices. Then you can get these adjacent matrices to construct a network. This is not trivial at all, okay? And if you work on voxel-wise analysis, that's the dimension is huge for correlation matrices, okay? And therefore, there's another level. There's typically two approaches that are common. People usually using ICA. One thing they are using is using ICAs, run it and get to the major clusters. And then you're using these ICA components to construct these correlation metrics. In that case, the dimension is much lower. Another thing we are doing is, or the other people doing is choose appropriate to the template. For instance, we choose there's a very famous template called Glacier 360 that's on the cortical surface. And the paper published in Nature 2016, something like that. And also we're using that as our template to do this kind of thing. In that case, the correlation matrix is much lower, maybe just like 360 times 360. Then you can do these kinds of adjacent matrices to threshold it. And then you can do the network analysis on that. I think there's the narrow, also there's one thing besides, there are two types of the correlation in generally. And one is just to calculate the correlation type of or some other kinds of correlation, basically, and based on some metric. And another thing that people are interested in is doing partial correlation. Basically, they want to calculate the correlation among two regions, condition on some other regions in different network. And they want to see how does that well perform. Well, that's one also is a no consistency. And because when this number of RIs is large, actually calculate the partial correlation itself. When your number of the time points per subject is relatively small, this partial correlation is also a disaster problem. Okay, it's difficult to do. How to do the partial correlation correctly? There's no golden standard at this moment. Or existing statistical method doesn't really work well on this. And I know there's many people doing this partial correlation stuff, but at least according to my knowledge, none of this method works. And on this function fMRI partial correlation calculations. And then you basically, then you get the adjustment matrices. One thing is some people doing binarize the adjacent message into the 0-1 kinds of the correlations, the network, and also some people just direct work on weighted network. And I think the one critical thing is associated with binary network is that's associated the number of time points you have, and it's not a trivial to threshold the data. Okay, I think that the most of the difficulties besides what we are looking at in the step 1, step 2 is also critically important. You know, you really don't know how to do it correctly, and also whether the findings in itself is really interpreted from neuroscience perspective. It's not that trivial. There's no consensus on this. People are doing that. That's fine. I think we're doing the same thing. But that's, I think that these 2 parts, step 1 and both step 2, are very important. Because my personal philosophy, garbage in, garbage out. I think that's you need to spend more time on the downstream analysis, in particular at the lower level. Okay, and some people are using the weighted network analysis, but how to do it correctly. And it's not that there's books and papers on doing this and using different modeling. I think that's fine. I think there are tons of opportunity over there, but it just you need to justify why should we take the approach, how much we trust it. Okay, there's many people are doing these kind of things. Yeah. For network constructions, you need to define the networks, and some of the key personalization issues in the contents of generative network modeling. You can use the anatomical attendance, what we did, and also you can use the data-driven personalizations, and you can use some of these like GLM type of things, results from the literature to define the personalization. And also you can use unsupervised learning method, like clustering method, like ICAs in particularly, and to do it. And also you need to define the edges, and the edges construction is also not trivial at all. And each of these kinds of steps is basically has tons of the uncertainty, and also they contribute a lot to the quality of the data. And one thing is people are trying to incorporate the structural diffusion trajectories. That's from the diffusion tensor data. And with this connectivity from the resting functional or functional fMRI data together, and then they can construct either binary graph and a weighted graph, and try to integrate the information across different network data, and to help you to construct it. Okay, that's construction procedures is not trivial. Okay, you can easily do it, but it doesn't mean that it really works. And also, there's a different metric you can choose. You can choose correlations. You can also choose some linear association metric, or the nonlinear association metric, like mutual information for detecting independence. And also the modular network, you can use the Bayesian net, multivariate autoregressions, dynamic causal model, dynamic connectivity regression, penalized regression. And you can see all these different tricks inside, but to be honest, people still practically never really using this kind of things. I personally don't do this much. And also the thresholding is difficult, and because you wanted to read, the reason you want to do thresholding is, so one of the key reasons is just try to remove the weak connectivities so that you can produce the adjacent matrices. Okay, and that's an open question. So credible network that require carefully choice of the thresholding approaches, and if we like the density of the connections and the network topology, they can easily yield erroneous conclusions. Okay, And there are three categories of the threshold message, include the fixed threshold, the fixed average degree, fixed age densities, and all of these things are attribute. You know, you can write a paper, methodology papers, that's for sure. But whether biological is meaningful, we don't know. And another thing about step 3 is about nanocomp modeling. You can use a descriptive models like graph theory and the complex network theory that come from applied math and physics. And that's the most popular method people usually do, just because they usually need to calculate some summary statistics. But most of these network graph theory and also complex network theory also depends on binary network. That's the reason my personal, uncertain and kinds of resisting on using this method, mainly because it's harder to justify. Okay. And also the reproducibility and whether these measurements from the graph theory are highly reproducible or not. But before the UCAT bank, we won't be able to validate and calculate the reproducibility. But nowadays we can, and that's one of the things that we're working on these days. Just wanted to see reproducibility on that. And also you try to do modeling and inferential methods like random graph modeling. And in particularly, you can work on this weighted network or this binary network. Either way is fine, but to me, is now this existing method really make a big impact in the field. And I think the graph theory is still dominant method, and the complex network because they're easy to interpret. And but it's hard to this is basically there's a tons of paper topic. Okay, you can write the papers, but doesn't mean that this. Yeah. Any questions regarding this part? But this is one of the hardest topic in the functional memory analysis. Because the data structure is complex enough, and there's many people working on it, but there's all these method magic issues. There's nothing wrong with the people who develop method. This is wrong because the reason mainly come from the data itself. Okay, you want to make a huge impact in the field because there's a lot of uncertainty. And also we're talking about diffusion tensor or diffusion weighted image analysis. And this is another major image modalities in the field. You need to have the big pictures of this. And this used to be a very hot topic. And there's still many people working on that. But actually, I think this field, and just like function fMRI, after more than 10 years of development, whole areas is more like a flat, okay, and move very slowly these days. Okay. Was there a question? Yeah, I can share some of the reference. There's several review papers, pretty nice. And actually there's several papers in the Neuroimage. And also there's a paper by my friends. And Simpson, they write a very nice review paper on that. Yeah, I can share some reference on this. Yeah, the people are working on it, but just I didn't see not many people using that. We are using the same thing, but I'm not convinced. Okay, but currently the lateral analysis, they originally they calculate the global kinds of the graph theory metric. Okay, these days they doing locally graph theory metric. That means they because the whole time series is cut into locally. And to try to do the things, and because you add another dimension to your the network metric, but that's okay. You just write people. I personally I use it, but I'm not a very fan of that. One now the key reason behind it is I think there's no justification or validation on this type of stuff at this moment. I have to, I did have some students work on that a little bit for several years, but the more I work on it, I step back a little bit. Okay, I'm not saying this is not a great topic, but I think that because at the end of the day, you think about this, why should we care about network inference? Still is the most fundamental questions in the neuroimaging data analysis, because our data is really about connectivity. Regardless you're working on functional connectivity or structural connectivity, you really want to merge them together to give a coherent story on that. Just I personally don't think the way we are currently doing is quite right, but it's good try. You can always try to do that. In particular, I don't trust anything related to just threshold of the binary stuff. That stuff is, to me, is there's too much noise. And why do we care about wide matter connectivities? Because nothing defines the function of the neurons better than its connections. Okay, that's a very important message. Why this diffusion weighted image become a standard. What is the wide matter? This wide matter is just like the gray matter is like CPUs. Wide matter is just a network. Okay, just all these kinds of blue lines connecting different, the nodes or the GPUs, the CPUs in the server. Okay, your brain is a very complex service inside, and then you connect all the different parts together. Okay, and but you keep in mind, this is because as we said before. This is you're looking at the two voxels in the brain. This one is in gray matter, another in white matter. Okay, this is for the gray matter. The gray matter, you see something the neuron, because most of them are dendrites, and actually this is basically all these axons or sheet. Okay, and because you don't suppose there's one single axon. You're only supposed to have billions of axons in one single voxel, because the voxel is a huge scales relative to the axon size, neuron size. Okay, and what can the diffusion weighted images do? Actually, the diffusion weighted images are able to looking at how does a water molecule diffuse in a particular areas. Okay, and that's basically the key. Okay, basically, if water molecule moving in the gray matter, they can randomly move. Okay, and white matter is not, because there's the pathway or like the highway over there. Basically, some direction you move faster, some direction you move much slower, because they have highway system over there. But here, there's no, this is a complete open air field. You can do whatever you like to. There's no traffic, whatever it is. There's no kinds of construction over there. Okay, and that's basically this is how do they get to the data. Okay, then basically, white matters. You see a lot of this kind of fiber bundle stuff. This is more like highway systems. If water molecule move on it, and they could move very fast if along that highway. Or they can move extremely slow because they want to crossing the highway. Okay, that's the basic, because that doesn't allow you to do it. Okay, because these kinds of infrastructure. But for the gray matter, because it's open field, you can do whatever you like to. Okay, and the basic random-wise, one molecule can move any direction they want. That's the reason you can see these kinds of signals differences. And you see these isotropic motions and anisotropic motions. And this is basically what is white matter is about. And this is you wanted to reconstruct these white matter bundles. they essentially, you think of the each wide bundles is the highway systems in the LA, okay? And this is basically this about. The car is much easier, much faster, smoothly along the tracks, and these kinds of highway systems. And why we care about these things, you think about it going back to the neuroimaging data analysis, and we are really care about 3 levels of the connectivities. One is anatomical connectivities, okay, basically a pattern of anatomical links. Given our current technology, what kinds of the anatomical connections link different brain regions together in what sense? And then we need to have a roughly picture. Second one is the functional connectivities. That's the spatial dependence, statistical dependence. These are all about correlation, partial correlation, or other metrics. Last one is effective connectivity. So we want to know the causal interactions. And that's the basically related. That's why we think about that's very tough questions. Because when you do, that's all the question related to how to design the function fMRI experiment to trigger some complex the brain network so that you can see the causal relationship on the different brain regions. That's the most difficult part. Okay, and all these things are important. You know, I think that the main problem is because the current method it doesn't really. It's not just about method itself. It's still also related to the data itself. Okay, and then make it harder to justify many things. Okay. And the brain network referred to AC-FC effective connectivity between distinct units within a nervous system. When you're looking at the functional data, you always need to think about this 3 level of these network metric. Okay, that's important. Okay? Otherwise, if you think about it just from statistical perspective, that's lose of the foundation of your data analysis. Okay, that's critically important. And also the units, and this is critical for you to elucidating how the neurons and neural networks process information. Okay. And that's also components when you do this kind of analysis. When I do these kind of things, I usually always think about it from that angles. The units include the individual neurons, and then in that case, you may want to need to get the data at the neural levels. Okay. There's another level. The function of fMRI want to provide that detailed information. You need using other image modalities to do it, and also neuron populations, and also the anatomical ROIs. Okay, because there's for the imaging field, and there's a lot of development going on this the neuron the brain connectivity at the neuron levels. Okay, and one of my friends, you know, he worked on this topic. The last year, Nature has a special issues on this topic. Okay, but they are not quite really functional of my MI data set. But that's the problems they targeted to. Okay, the very low level neural levels. And this is very important. At least the diffusion weighted images provide the partial of these kinds of network analysis information on that. You don't suppose that one image modality can do everything. Okay, that's not the case. And you need to think about all you can think of. Just like the slimmer data is all these jivas is not a single. Just to give you one aspect about genetic information. And all the imaging modality is the same thing because each imaging modality have these strengths and they have the weakness. When you really think about something deeper, you need to really integrate all the information together. Okay. And now acquisition mechanisms and how does the diffusion weighted images doing? And the key things is we try to map accurately the structure and orientation of the fiber tracks in the wide range of the human brain in the vivo. Okay, and to be honest, with each location, each also they just estimate this 3 times 3 matrices, okay? Or a multiple mixture of these matrices, okay? But most of the method is either just as one single 3 times 3 matrices or two at the most 3 3 times 3 matrices. Okay, this is a positive definite matrix. Why this is a positive definite? And actually this is related to diffusion process. Okay, it's related to a Brownian motion. How does the water molecules diffuse in areas? Actually, we are interested in the coherence matrix of this diffusion process. Okay, that's the key. And if you in locally there's no barriers, there's a free diffusion is isotropic. Okay, and that diffusion is the same in all the directions. Okay, and then in that case you can characterize the diffusion process by a single diffusion coefficient lambda time the identity matrices. And but if you have this barrier to the diffusion, like you have wire matter bundles over there, and actually that's the basically you can estimate the coherence matrix like this. They become an ellipsoid, and there's some key directions, the principal directions. That's the basic fiber boundaries. We just use these 2D cases as an example. You can easily do it in the 3D cases. Okay. And then therefore, if you're able to construct that, then you basically get to the same ideas. And in practice, you basically at each location, you wanted to know what is isotropic diffusion or anisotropic diffusion. What is going on with this is basically when you have these kinds of two isotropic and anisotropic locations. And actually, when we do experiments, we're using radio frequency to force the molecule to move along one direction. I just impose the frequency pulse, give the energy to the water molecule to run to certain directions. I ask them to run to these directions, okay, another directions. Basically, when you do it, basically we run. Now this water molecule we force the water molecule to move to certain directions. And in the isotropic areas, because your force is the same, and basically you didn't see the differences if you force the water molecule to move. But for the anisotropic, when you force the water molecule to move on a certain direction, some part is basically move faster. You'll see the signal drop dramatically. For a certain part, you cannot move at all. The signal doesn't drop. That's tell you the differences. Okay, that signal difference tell you the coherence matrix. Okay, because I force the water molecule move along certain directions. And this is basically the faster the diffusion is, the more molecule change their positions. And that's the perfect refreshing become near to a signal loss. That's the basic tell me the direction information locally. Okay, that's all the diffusion weighted image about. Okay. Then generally, in the diffusion weighted images, you have the signals without the gradient application. That means if you impose gradient, that means I usually impose the strength or like the energy to the water molecule, and I usually give the strength or directions. Okay. And this is basically the signal noise equations to characterize that. Okay, we'll go back to this later. And this is basically then when you're looking at it, you get to connect to the data, and then you can basically try to estimate the diffusion, the coherence matrix of the D for isotropic or anisotropic. You get the coherence matrix on that. Okay, that's just to give you a simple example. And this is basically the math equation behind that stuff. And actually, for diffusion weighted images, and this is still a Fourier transformation of the signals. The EQ is the observed signals. Q is the direction. That's the energy direction. I enforce signals to walk along that Q direction with a certain degree of energy. Okay, that's the amount of energy I give to you. and also the direction I force the signal to water molecule to move. And then you basically write it as the Fourier transformation of that. Well, this is basically the P is our target, because we want to construct locally is an ellipsoid or the more complex shape. These are the kind of, that's the type of signals we're working on, okay? And this is the additional, like the signals and this additional constraint we impose on it. This is just to tell you, if you force the water molecule to move one direction and the opposite direction is the same, because that's the big, because our signals does not have this like direction information, just the along these directions. You can go either going up or going down, that's both fine, okay? And this is the basically diffusion tensor images. And this is the EBs observe the signals. And basically we try to, the U is given, B is the strength of the gradient. The D is the Coherence matrix. That's the reason you observe the E, U, B. This is three set of the variable, the observations. You measure them multiple times, and then you can estimate the D matrices, okay? That's the Coherence matrix of the diffusion process about, okay? And this is basically related to the U, you need to force the water molecule move in the space, on the sphere, okay? Coordinates, that's basically how many directions you should have. You can do the 12 directions, 42 directions, 120, 62 directions, 60, 42 directions. And then basically usually we do the uniform sample on the sphere, okay? And this is basically there are several key things here. The one thing is the B values. Another one is the number of the directions you apply the data. You acquire the data. Okay, that's basically the key things. And this is the DTI statistics. And for DTI statistics, there's the several key components inside, very similar to the function of MI. But in general, it's different, okay? Because the data is more complex in a certain degree, okay? And this is basically your first thing is how to do the attendance building. It's not a trick at all, okay? And you do these quantitative cartographies and the brain network analysis and predictions. Okay, that's basically the DTI statistics about. And the first things, these are the key steps, okay? Beginners, okay? And what do we teach? From now, basically, you have this raw diffusion weighted images, okay? That's the imaging data you have. At the same time, each time you know the P values, okay? That's that's related to strength of the gradient, and also the direction, the direction of the gradient. And basically then you need to reconstruct the diffusion, the D matrices at each location. Okay, this is basically the first row is the raw data. And the second row is basically the first one is the ellipsoid constructed at each location. And this is the principal direction of the like ellipsoid. After you get the ellipsoid, then you can using the principal direction to doing the fiber tracking to get these kinds of fiber tracks, okay, the wide matter tracks. That's how do do this imaging analysis from the raw data, reconstructions, and then your principal direction, and also doing reconstructions. And the second thing is doing atlas buildings. And there's a different way of doing atlas buildings. And one thing my friend do is basically they calculate some invariant measures like FI values. And then you're using the standard, basically the retration method to do the atlas. And then you're doing retration. But I personally don't like it because I think this type of method ignore these kinds of the tracking, wide matter track structures inside. It's not very reliable. Okay, I personally don't like the way they are doing it. I have my own way doing that. Okay, because I'm pretty familiar with this whole topic. I know how to do each of these steps in very low level, and also doing statistical analysis. And when you have this atlas built, you can do the voxel wise, and also you can do in the skeleton wise. Okay, this TBS is the skeleton. This information you can project some of the environmental measures and on these skeletons and do compression. Or you can basically using the white matter tracks and project the information on it and doing the functional data analysis on that. This is basically the three key things on doing this. Okay, basically you know how to reconstruct the data and doing fiber tracking, and you need to know how to build an atlas and doing statistical analysis on that. And actually, this is my own way of doing this. And we have a paper published in 2018, Neuroimage. And I think that this is one of the best methods in the field. And we basically we're doing tracking, pixelation, fiber counting, FA extraction to track compressing, FA compressing, and doing this lateral work analysis. Okay. And we have these kinds of whole pipeline from beginning to the end. Okay. And any questions? And just to tell you what's going on with the noise models, how to do reconstructions. And the key ideas, you can read my paper, published in 2008 in JASA SES, where I have a discussion paper on that. It's basically is about a single tensor models. This is the S is the image intensity. S0 is the baseline images. You have to estimate it. It's more like intercept. Then you have this B is the strength of applied gradient. G is applied gradient directions. You observe this data, SI, BI, GIs, okay? And you estimate the Ds and S0. In practice, you need at least to have two different B values. Otherwise, this model is not identified. The D is not identified. You need to have two different B values. In practice, usually they choose the B equal to 0 and something and another relative large B values. Okay, and they can estimate S0 and the D. Okay, and this is basically the model. In my JASA paper, you can read it. And this is basically the regression models, and you can using region noise, or you can using regression models. You take a log on both sides, and you can get something like that. And this become a semi-parametric models, and you can basically construct an estimator for S0 and the D. Okay. And you can use this weighted NESS square estimator, but there's what I did in my JASA paper. First, let's start a NESS square estimator and then estimate the weight. And that's the weight is mainly come from the noise, this part. Okay. But you can just take a look at the weight, and then you can create a weighted noise and iterate for a few times, then converge. And after you get that, then you have this diffusion, the T matrices, then you can perform the singular value decomposition. You can get a principal direction and also the first lambda. Okay, that's the basic most important parameters, eta 1. Okay, and also the 3 eigenvalues. And after do that, then the relative relationship between the lambda 1, lambda 2, and 3 tell you the shape of a local ellipsoid. This is spherical and discoid, pronate, and non-singular. And in practice, these two shapes are very important because that's C and the D are important ones. And then these also have a different environmental measures, like FA, CL, CP, ADC, and the radial diffusivity. There's all these different environmental measures associated with eigenvalues. And practically, people use these parameters, these environmental measures a lot. Okay, and this is basically, and they use these environmental, calculate these environmental measures at each location, and then they can use that to do these kinds of clinical, they can use in clinicals to compare the different subjects. And this is the mean environmental measures are commonly calculated. If you are familiar with the literature, you see all these kinds of terminologies surrounding around, come up all the times. And these environmental measures are quite important. And this is basically, they tell you the several scenarios, the high number of the fibers, the high diffusivity, and the high FA, no diffusivity. This is the myelination of fibers, the high diffusivity, no FA, but the high FA, no diffusivity. Size of the fibers, this is basically high diffusivity and no FA, high FA, but no diffusivity. Directionality of fibers, and no FA, same diffusivity, high FA, same diffusivity. This is the reason in practice, people usually don't really work on the ellipsoid itself, the D matrix. They're always looking at some principal directions and environmental measures. Okay, because clinically easy to interpret. Okay, and any questions so far? And topography algorithms used to be one of the hardest topics in the field. And they move very fast, but they slow down these days because the problem gets harder and harder, and it's very difficult to do. And this is the white matter bundles, and basically, and how to. There's one of the popular algorithms is the streamline tracking algorithm. The key ideas is at each voxel you calculate the principal directions. And that means your non-local tangent vectors, okay? And then you're using that one, basically try to see that we just using a starting point, and then we write fast algorithms, basically connect all these points, voxels together to make these kind of the fiber bundles. Okay, they can run very fast. Okay. One of my friends wrote a very efficient algorithms and to simplify these problems. And this is streamline tracking algorithms. What it is? This essentially is very interesting things. Okay, this is related to. I'll just show you some examples. These are the in vivo analysis of fiber trackings, and basically, and there's several major fiber bundles. And then how does the white matter looks like by using the diffusion weighted data. Okay, and there's a lot of development in the early 2000s. Okay, but all these things are related to the following aspect called the stochastic diffusion equation, the differential equations. And some people, statisticians, are working on, did some work 10 years ago. Yeah. And but the key ideas is this is basically the problems that we try to solve is stochastic differential equation problem. But the reason is at each the VT is the fiber track curves in 3D space. Okay, this is the problem is harder than most of the non-parametric problems in statistics. Okay, and because you don't really observe the VTs. Okay. And what you observed is you only observe the tangent vectors. That's the principal directions. And actually that principal direction, the E hat, but you can get that one from the diffusion weighted images. And you want to recover VT, then plus noise. And basically you're trying to solve this noise version of the differential equations. Stochastic differential equations. Okay. And this is basically the kinds of inverse problem we want to solve. Okay, it's not trivial at all. Okay, and you can do a lot of deep math on this. And this is basically the how to do the tracking algorithms. Okay. And this is the analysis framework. And there are three major methods. One is called a regional-based method. One is a voxel-wise method. Another is a quantitative topography method. And the regional method, basically you'll have the attendance, then you'll do everything in the ROI-based method. But the people gradually get rid of this kind of method because they think that take average within each ROI for the diffusion tensor images is not quite right way of doing things. Okay, and then they doing this correlation to the attendance. The question is what kind of NS we should use for diffusion wedge images, and it's also not a very trivial problem. Because for white matter, you have these kinds of the white matter structures. How to do the retracement correctly is not trivial at all. And I don't believe anything people have done so far. That's not a problem. And because simply because your data have tons of structure inside, the kind of algorithms itself is not flexible, good enough to capture all these information. And the voxel-wise and other things, people get rid of it in the literature, in the sense of because we don't think of the current retribution based on any diffusion wedge the signals features are valid, so that the most of the people just don't. You cannot get a paper published for doing this. Okay, and all these things involve the study specific attendance doing. Another one is a TBSS. This one is the key ideas is the determining the white matter skeletons, then map these FA values onto the skeletons, then doing voxel-wise analysis on skeletons. And this paper published in 2006. This is one of the most popular methods in the field. Okay, the key reason is the skeleton is pretty robust structures. And even though it doesn't mean that skeleton is corresponding to white matter bundles, but it's very robust. It's easy to implement it, and then it's easy to reproduce it. Okay, that's the reason this method have almost 10,000 citations. Okay, and because people using these tools, this is one of the key reasons. And then you can just doing the data analysis on that, and the voxel-wise method analysis, simply people abandon that. And another thing that people have been trying is doing track-based analysis. And basically, you're doing track. First you're doing tracking, and then you're basically doing all the analysis on that. And what the problem is with the tracking itself is highly depends on whether you're able to faithfully track doing fiber tracking. Okay, that's one of the key technical barriers for most of the data. In particular, if the data is not high quality. Okay, and our group is one of the key players in this topic. You know, but that's we try to solve these issues. They are trying to model the fiber tracks. When you do the fiber tracking, then you can project the data on it. And so that you can basically, like FA value on it, they can do all the analysis. These are fiber tracking in the attendance space, and then you can do the comparisons across that. And this is the transfer of the fiber to cases, and then you can basically doing comparisons on this. And this you can do the fiber profile analysis, and that's one of our papers published in Neuroimaging, and basically how to do the functional data analysis on that. And this is the pipeline we developed, and you're welcome to use it if you want to. But in general, this is basically the three image modalities: structure, function MI, the structure MI, function MI, DTIs. I try to use almost 1 or 2 hours just to cover the whole things, big pictures, just so that you know that for image and for these MI images, what kinds of major analysis kinds of pipelines people have been using on that. Okay. And any questions? At the NESS, I think you have the big pictures on this, because each topic is a text. I can easily teach each topic for 1 or 2 days. Okay, but I think the key ideas is already there. You're at the NESS, you have a roughly idea about how much people can do in this area. Okay. Next, take a 10 minutes break. We come back in the 2:55. Can you see my screen?
Yes, yes.
I think we have another 2 hours, right?
Yes, to 5 p.m.
Yeah. Because this, underneath in the last lectures, I cover 3 major image modalities. One is the structure MRI, another one is the function of MRI, another one is the diffusion weighted images, DTI stuff. For each image modalities, there are some fundamental problems inside questions people need to solve. Okay, like for the structure MRI, what are the most important questions to me is how to faithfully, reliably to segment the cortical, subcortical structures, okay? And then you try how to quantify the data for that, okay? In the sense of how to really map that data into a common space and do the comparisons. The problem is not that trivial, okay? Even though the current have some, there's a lot people develop some tools for that, but that tools still, have some many issues. Okay. And for second part of function of MRI, the key things is, and it's mainly related to network analysis. How do you do the network analysis right? How to map the signals to a common template. What the atlas should look like, okay? And there's no conclusion on that yet, but there are a lot of progress on this, develop the better kinds of the atlas for function family data, okay, for brain functions, based on function family data. The third one is mainly about diffusion weighted images. The key challenges are related to how do you really build a white matter kinds of based atlas, and then you project your data on that atlas. And there's also a lot of progress made so far, but that's still this kinds of, in my vision, the next several years, what the whole field should look like, okay? And at least you'll have some basic ideas, how for each image modalities, then you can easily just start to look in the literature. So you can focus on one or multiple topics you can work on. And we are more to the population level image analysis. I can easily talk in 2 days or 3 days on this topic because there's tons of results on this topic. But as I said, the many topics is emerging ones, and because we only are available have this type of data recently, in particular large scale data sets. And I cannot, because we only have 2 hours left, and I cannot do too much. Okay. And actually, you can watch my videos on the website. And because I think in the YouTube, there's one of the talk I gave in the University of Michigan last year. It's about imaging genetics stuff, mainly related to clinical findings. How do we use the data set to do the imaging genetics? And it depends on how much time I have, because it's only 2 hours. I want to be able to cover all these 6 or 7 major topics we have been working on. And also I'm trying to write a book on this and just get too busy these days. I should be finishing pretty soon. And I think we'll get these review papers ready in 2 or 3 months. But the book, you have to wait for another year. But we cover all these things that we discussed today on that. And I just give you a quick through what I did for this topic is on these object-oriented regression models. And there's many papers published in this topic. And I just don't go through all the details, and just give you some roughly ideas of these things. And the object-oriented regression analysis usually will refer to the some complex objects, okay, and like the functional data, okay. Like the brain imaging data usually is the functional data, and also we refer to the like a matrix data. Okay, you'll have the connectivity how to analyze the matrix data. We're also talking about this other the network data, and there's all these different object or like shape data and how to analyze it. Okay, I will go very quickly on these things. Okay. And this is the key features of the imaging data. This is most imaging data is infinite dimension, have strong spatial smoothness and the spatial correlation and spatial heterogeneity. Okay, and this is basically make these problems challenging enough to develop the method. And this is the basic. We start at the beginning. The imaging data is infinite dimensional images. Okay, that's the basically. When you're looking at the images, you see you see clear pattern inside. That's because the data is relative kinds of simple on that aspect. And one thing is we how to characterize the spatial smoothness. Okay, and actually we took this one of these kinds of spatial smoothness from the segmentation, or even from the computer vision. We call the cartoon models. That means we divided the whole regions into the multiple disjoint areas. And then we basically, within regions, we assume this piecewise smoothness assumptions and the smooth boundaries. And then we can do this local patch. And then we allow these different, and there's a jump around the boundaries, okay? But within the partitions, each interior of each partitions, this basically will allow you to be smooth. Okay? And these are the typical models people use in computer vision and imaging analysis. And this is the basically we call the cartoon models. And this is when the simple is the case, they assume this kinds of the is a piecewise smooth the functions with discontinuity along the smooth the hypersurfaces. Okay. And also we try to characterize the spatial correlations. There's two types of the spatial correlations. One is the long range correlations. Okay. And the usually is the long range correlations mainly come from these unmodeled effects. That's because the cohorts you include in the models is not is not large enough so that you cannot really model some long-range correlations. Okay, and another also we also try to model the short-range correlation in the sense of this local smoothness. Okay, this is basically there's two types of correlation structures inside. This one is related to spatial smoothness. Okay, this one is related to spatial correlation. And I'm not going to these details with the functional data analysis like random elements. You can just take a look at the papers in the field and doing that. And then we have this basically in practice, we have this smooth the functional data. I will try to correlate with the chlorides. And also the typical example diffusion, DTI, the fiber tracking data. And we have this diffusion properties along the tracks, okay? And this is basically the math behind it. And I won't go to the details and then another thing is about functional PC. I won't go to the details. Yeah. And I just show something, show the cases. There's one paper we published in in 2011 and 12 is try to model the multiple functional responses. And how to do it. And this is the basic of the model we write. This is your response, okay? And this S is a spatial location, okay? And the K is a different component, right? This kinds of, this is the covariance effect, and then this is the functional, this eta is trying to model the long-term non-range correlations, and this is the epsilon is shorter range correlations, it's local correlation structures. And we have these different covariance. And we assume this BS here is kinds of smoothed functions, okay? And this is basically, you can easily construct estimation theory on that. And this is basically, you have these kinds of the symptotic normalities results, and also you can easily do that. And then when we do this stuff, the key things is we smooth the data first in order to reduce the contribution from the short range of noise. And that's the typical people doing the functional data analysis. You smooth your data first, okay? And that's what is the results about. And then you can smooth the individual functions, estimate the coercion operators, you estimate the eigenfunctions, and then you can do the functional PCR, okay? And you can have a sequence of the steps for combine them. And also you can test the linear hypothesis and basically to get to the local testing statistics and the global testing statistics, and do the hypothesis testing on that, okay? And this is the basic symptotics, and you can have the critical points and the confidence intervals. That's all you can find in the paper. We want to go to the details. And this is basically the input informations, and you can run through all the things and to get all these suggested inference tools for that, okay? And this is a real data analysis we did in the paper, and we have 5 diffusivity outcomes, and from this 128 subjects inference, and this is basically fiber tracks, and also the different environmental measures on these tracks, and then we can do the analysis on that, okay? We have many interests in gender and also gestation age effect. And this is basically the result. You'll see that there's a strong gender effect, age effect, very low gender differences across, okay? And all this is confidence interval spanned about. And this is the function of PCR associated with that, okay? And this is another paper we published, is a functional MEM. And this is a functional mixed effect model. This paper published in Statistical Cynical, I think 2 years, 3 years ago. The key motivation is about, we have this longitudinal track data. Okay. You see that this is the same subject, FF values along the fiber tracks. And actually these 2 weeks of this, the 2 weeks, and this is another, well, this is the FA, this is the tensors. 2 weeks, this is 1 year, and 2 years. Okay, there are 3 major fiber bundles, the same fiber bundles across time. Okay, and then we develop these kinds of the longitudinal extension of the first models to these kinds of Y as T. T is time, and we add another dimensions. Then we can just feed these functional mixed effect models, and to basically to analyze the data. We call this a spatial temporal process. Our object is to dynamically to quantify dynamic functional effects of covariance interest on the functional response. And this is in these cases, and we introduce the additional kinds of changes here. You see that this epsilon d t, that's both spatial and temporal, This is to characterize local correlation noise. Key differences is basically we introduce both eta I d the t and also cosine ID. You see here, this is the kinds of key differences. We added this because the dimension increased, because we have additional temporal information on this data, so that we have to do it slightly different. Okay. And that's basically very similar to mixed effect models. I won't go to detail, just you can take a look at our paper on that. Okay. And this is the basic comparisons and with the existing literature on linear mixed models, my friend Gross method, and also Graven's method, and also our method. That's the difference regarding to the estimation and inference tools. And this is basically the pipelines for doing these things. And this is all similar to the first paper. That's just one of my students' work. We just write another. And this is the real data analysis. And but now we have these multiple scans per subject. And this is basically the neural scan only, the 1 year scan only, and also this is the combination of that. And this longitudinal data analysis, and that's what we did. Okay, and this is basically the curves relate to age 0, 1, 2, and how does these kinds of changes across times across. This is the real data, the motivating us to this development, and this is the real data analysis on that. And this is another paper is called the functional nonlinear mixed effect models. This is all because I'm doing statistics originally. This is the paper we did about 7 years ago. And what we did is basically we still model the similar things on the second paper. Okay, we have YIJS. This is the spatial location. We assume this is a nonlinear functions of f. F is the kinds of given except some parameters. And this is phi I is the fixed effect, okay? And then plus epsilon. We are model this kinds of hierarchical. We assume that the phi is equal to the beta s plus pi's. It's a running effect. And then you'll basically still want to construct asymptotical properties associated with this type of models, okay? And then you can basically, the estimation procedure which you do at each grid points and then merge it together doing smoothing. And that's how we can make a statistical inference on that, okay? This is basically tell us if you do it or don't do it. If you're using our approach, you can dramatically increase the statistical power for doing that. Okay. And this is the real data analysis we did by using the autism research data. And this is basically the number of subjects with one visit or multiple visits. And this is the models we use, compares functions. Okay, that's our starting point. The functional version is something like this. Yeah, these are the fitting results for our models. And you see that the data is pretty noisy. The fitting is relatively good. Okay, then you can construct the confidence interval bands. This the first of the 3 papers is the same lines. Basically, the first one is our original paper to do the regression, the statistical inference on that. Then we extended to longitudinal data in 2 different way of doing that. One is linear mixed model extension, and another is non-linear mixed model detections. This is all the work we did about 7 or 8 years ago. And another thing is I did with my former PhD students from NC State. And this is about the functional structure equation models. The second one is did with my former postdoc, the Benjamin Rich. And now he's assistant professor in Emory. And we did a paper published by Statistics. is the ideas is something like this. And if you are familiar with the twin structures, the existing twin structures usually, the twin models is you have the MZ and DZ twins, okay? And this is the standard of the mixed effect formulations. But you can apply these kind of twin models to each point on a functional data, okay? But the key problem is now, right now we are facing is we have functional data. And one question is I asked myself, could we just extend the twin models and from the univariate measurement or multivariate measurement to the functional data? This is basically the project about. Okay, we try to extend to establish twin models for functional data analysis. And what we did is these are the models we write this way, okay? And this all the functional, this AIJ is the functional data, the CIJ. We choose these representations because it's more natural to formulate this for functional data. There's some other representations in the trained literatures, but this one is much easier to handle. And you can read our JASA papers. Just we have a lot of detailed derivation from this. And actually, how to estimate the parameters. And we propose these two structures. One is mean structures. Well, first, the estimate the mean structures for this beta. And then we using some smoothing tricks to make the beta looks better. And also the variance structures, we subtract this stuff and then we check the variance structures. And also we construct this weighted likelihood to estimate the sigma and the sigma A and the sigma C. Then we estimate and related to the parameters, then we can get to these kinds of quantitative operators. We basically decompose the problem into three layers and do that. Then we can do the testing statistics for marginal and the environmental factors, and both marginally and globally, and for functional data analysis. Okay. And this is basically the two beta D and also the sigma A, sigma C, and this is basically estimated the functions and also the current structures. It looks pretty reasonable. And this is the simulations to demonstrate that and our method, the power and the null hypothesis, and how much we gain by doing our approach. Okay. And this is the type 1 errors for our method and the rejection rate. And along the fiber tracks, when C equal to 0.5, you see that our method is much better than the simple pointwise estimator, the testing statistics. Both, and this we can control the type 1 very well, and but for the powers, we're much stronger power on that. This is basically the data set that we use from John Gilmore data set because there's a lot of twins in the studies. And these studies are about 900 subjects, and there are about 356 healthy trained neonates with 190 males, 166 females. There are about 48 MZ twins, 81 DZ twins, and 89 are related singleton twins. And the question is how to comprehensive the heritability data on the white matter microstructures, FA, ID, and AD along these 47 major fiber bundles. And this is basically what is the sample two pair of the MZ twins, the two pair of the DZ twins, and we have this kind of functional data, how to do data analysis on that. And this is basically the FA, the intercept, and gender and the age. This MD and other event measures, you can see the estimators. And this is Covariance metrics, and also the estimated common environmental covariance for FA and MDs. It's pretty interesting patterns. And also this estimates the genetic variance along these fiber bundles, fiber tracks, and the associated with the p-values. Okay. And these are the heritabilities, and in these 47 traits, and we get. And one thing is, we tried to extend this method to the high-dimensional functions. In these problems, the data dimension is still relatively small because it's about less than 100 grid points in one-dimensional case. But here we are dealing with tens of almost 1 million points. How to handle that? That makes the life much harder visible for us. Then we tried to extend it to high-dimensional functions and how to handle that. Okay, and this is basically what we did in the second paper. And you will see that basically by using our method, both computation, the estimators, the mean square is much reduced if you treated the data as a functional data. Okay? And we apply to ACP the cortical thickness because ACP is basically twin studies. And we choose 556 subjects, and there's non-smoothing and also AMZ and 2TZ and 200 singletons. And we corrected some covariates inside. And this is the heritability estimators by using our method, okay? And this is around 0.15% to above. Okay? This is basically the two papers we wrote several years ago, okay? And you see that the first three, the one is from the functional data, second one, third papers, many about longitudinal data. This one is about twin data. This is the serious work we did along these directions. Then also we try to do this stuff is try to doing imputation, not doing imputation, is doing predictions, okay? And actually the imaging data, this is the big missing data problems. There are several things we're particularly interested in. One is missing imaging data caused by design. This is also very common in the genomic studies, because usually for set of subject, we only acquire a specific type of imaging data. We don't have measure the other kind of imaging data. What can you do about it? And for some set of database, we have all these imaging modalities, okay? And also we have the missing imaging data causing back positions because the hand motion, physiological fluctuation, artifacts induce problem, and susceptibility artifacts, okay? And this is basically that's what I say is about missing data problems. And, we are working on this problem right now. It's not that easy to solve. And you'll see these data structures. Sometimes you'll have these kinds of cross modality missing, okay, and also cross subjects and also a lot of local spatial smoothness inside. And we developed this kinds of message is basically to predict the particular models. It's still based on our first model, the very coefficient models, but it's slightly different. Okay, and there are two kinds of things we are working on. One is basically eta ij's. We use that to characterize both individual image variations and also media and to London range the dependence of structures. Okay, that's why do we introduce eta ij? We also impose smoothness the conditions and the beta j across the image modalities. Okay. And this is basically the model we built. It's very similar to the first very coefficient model paper we did. Well, key things in these cases, we're not doing inference, we're doing predictions. And actually we combine a functional PCM models and also a multivariate simultaneous autoregression models to improve the prediction accuracy. Okay, and this is the basically these estimation procedures for our method in the neuroimaging paper. Okay, and this is the basic simulation studies that show you the effectiveness of these kinds of spatial Gaussian process models. And this is the basically the first one is true. The second one is the estimated beta images. But this is the precise coherent structures for doing that. Okay, and it's true that our method can really achieve better performance. And also this is the real data we did. Okay, this left natural ventricle data from 43 infants. And this is the basic, the spatial data is something like this, okay? And this is the basically, we applied our method to get the get the parameter estimator as a function. Then also, yeah, this is the result. The eigenvalues, principal directions, current the functional PCI results, okay? And this is the comparison among the three methods. The first column is the voxelwise linear models. You see that the Rose mean square P is pretty large. And if we add a GLM plus the functional PCAs, and which much better than the first one, okay? But the last one, we basically use our method to incorporate both functional PCA plus autoregressive the structure inside, okay? First one, we didn't consider functional PCA. The second one, consider functional PCA. The third one, basically, we consider both functional plus the local kinds of other correlation structure inside. You see that the prediction accuracy is much better than the existing, the standard, the Paris method. That means, in practice, when you handle the neuroimaging data, is the first things you need to introduce two level or kinds of the spatial relationship. Three level, one is the spatial smoothness. And also you need to incorporate functional PCA to account for spatial correlations. And also you also need to do local correlation structures to capture the local correlation patterns inside. All these three kinds of structures in the data are quite useful for improve inference, accurate inference, and also the prediction. And this is another papers we did is in the data structure is something like this. Okay, for some data set we're missing one of the image modalities completely for one subject, for multiple subjects. Okay, and this is basically we have this longitudinal neuroimaging data. And this is the model we try to incorporate the temporal information inside, both the subject and the temporal information inside. Then we can model the data, something like this. Okay, it's very similar to longitudinal stuff, but we have this across the mu is across both subjects and time and across modality and time, and also we have local spatial temporal smoothness. And basically across both T and T. And this paper is published in also in the Neuroimage. And this is basically the same thing. We try to model spatial temporal correlation structures, and both by using functional PCA and also the local kinds of the grading type of the method. And then we applied it to the adenine pad data, and you see this is basically the occurrence structure that we impose on the local structures, and you'll see that this is the results from functional PCA part, and this is basically let me see. Yeah, one is the first row is the observed patch images in the testing data set. The second one is second row is predicted. And you see that the results are quite similar to each other in many cases, in many locations. And yeah, this is another set of the prediction results for 3 different subjects. Okay. And this is basically the root means SPEM images. And the first of the columns is the semi-parametric models. The second models is the semi-parametric plus the functional PCA. The third one is you add a functional PCA plus spatial temporal local spatial temporal structures. You see that basically the results are pretty consistent. And then you can see this Hongtu mean and MSPE method, the parameters for the 3 type of the models. And the conclusion is basically in practice, for all these messages we try to deliver here is, if we incorporate the spatial smoothness, that's the one thing you need to consider. Another thing is you need to try to model the non-range correlations and also local or medial to local correlation structures in the data, okay? And this is another paper we published at SBCM, and this is basically, we try to model the piecewise functions with unknown jumps and edges. Okay, this is the basically type of things we will try to do it. And compare with the previous papers, we're always assuming that the beta images and also all these functions, the coefficient functions are smooth. Now we allow the coefficient function to be piecewise, the noisy piecewise smooth functions, okay, with unknown jumps and edges. And this is basically what we did here. And what the key difference is we are assuming that the BD here, the coefficient images are piecewise smooth varying coefficient. Okay, it's not a complete smooth. If you piecewise smooth, when you do that, to make the life much harder to deal with. And then we would try to incorporate the cartoon models for the coefficient images. And then this is what we did. And this the key ideas. So we tried using kernel-based smoothing method and to do it. And there's the different type of the kernel-based method. You see that f hat equal to summation of the omega yj's. There are several different popular kinds of weights like local constant, not local linear. Also, you can using the Yaroslav-Skalevsky and the binatural filters, or non-local means or propagation separation method. And this what we did is we using our propagation separation method. The key idea is each voxels we can start a sequence of the bandwidth. Then we can at each from the low scale to larger scales, we estimate the weight and estimate the estimators. We have these adaptive weights adaptive estimators, and then we basically do it local. And then we have these adaptive smoothing coefficients and also estimated coercive operators and the estimated argument. That's all the other things are very similar to the way we did it before. But the only difference is here we are showing these kinds of the cartoon models for the estimated images. This is basically give you a simulation studies about and the true images and the SVCM, the developing in this paper, and also the initial estimating SVCM and all other methods. Okay, this is the. You see that our SVCM method is much better than other methods, and there's a bias in our method, and also bias associated with other methods. Okay, and for different the bandwidths. Okay, you see that our method performed pretty well in practice. Okay, and this is real data, and that's the ADHD data set, and we are using 174 subjects. I told you this paper all finished almost 10 years ago. At Denise almost 10 years ago. And that's the reason the data set is pretty sample size is pretty small. And also this is basically the interaction effect for H. The H0 is no smoothness, H equal to 10 NESS strong smoothness. This is basically the gender and diagnosis status, okay, for that. And the 4 eigenfunctions, then we estimate it. You see the major pattern in these eigenfunctions. That means there's a lot of the noise, we cannot explain it in the data, okay? And also we observe that the smoothness does improve the prediction, the estimation accuracy. And this is also associated with the regions associated with the effects, okay? Yeah, this is basically a set of work we did for the functional data analysis, okay? What we did is basically, the lesson we learned so far is we started with SVCM, and then we just extended it to the longitudinal, and also try to incorporate smoothness and the piecewise smoothness and the functional PCA, and also we plus this locally the kinds of spatial temporal correlation structures. And this really depends on our purpose is doing estimation, or the purpose is doing predictions. We do find that when you properly to model the data by incorporating all these information together, and then you do achieve much better performance in practice. And I think that the following, I just give you a set of the things we develop for metrics. And if I'm interested, there's a series paper we include here. And this is the metrics to value the data. And the first paper we developed called the low rank linear regression models. And this is basically the different data types and different studies. And the key things is we wanted to develop method for imaging genetics, okay? And we have the genetics, imaging, and the disease, okay? We try to model their relationship. And we have several papers just basically focus on that. It's just this is an imaging field, and you can do much better than that. And and we started with this kind of hippocampus surface data, okay? And this is basically we represent each of the hippocampus left and right as some matrices, and each of the element in the matrix represents of the sum of the shape information, okay? And this is basically the hippocampus surface data, okay? That's the shape data. And we try to correlate with genetics, and this is basically there's about 6 million of the imputed and also quite SNP data. And the question is how to correlate with this hippocampus with the SNP data, and how to do the GWAS on that, okay? Because this is, you think about it, the domain is huge. You cannot just run this 100 times 150 and treat each of element as the phenotype. That's too time consuming. We won't want to do that, okay? And given this scope of size, and this feed a gigantic models accommodate all the genetic variation imaging measures, and it's simply is optimal memory, okay? And another approach you do is you fit a univariate model to each of the voxels or each pixels and also each SNP pair. Then the number of models you fit is also big, okay? And we wanted to do something between. We don't want to feature voxel-wise or pixel-wise GWAS analysis. We don't want to really put all the data together and run a simple model. That's not the right way of doing things, because we simply don't have the memory or the resources to do that. And this is basically what my former postdoc, Hong Kong did how many years ago? I think because he almost get a tenure at the Toronto. That's at least 8 years ago. We started 9 years ago. We started this project, and this is the basic the model we try to feature the data. The yi is the matrix, xi all the SNPs. Okay, this is the model we started with. Okay, but at the very beginning, we don't know how to really model the data and try to come up with something. And what we did is we finally it goes back to why don't we just doing some screening and estimations. And then the first thing is we just pick up some simple ones using sure independent screening method to do it. Okay, I just show you some trick. And the second step, we refine these estimators for the significant queries and develop a low rank estimation procedures using the nuclear alarm penalties. And it seems like this is very straightforward, right? And it turns out it's not that technical, straightforward. And when you try to screening the whole matrix, because your Y is a very large matrix, it's 100, almost 150. And also your X is univariate, just in the sense of you have all these SNPs. One thing you can easily calculate is ordinary square symmetry. Okay, for each SNP you can calculate the B hat. That's the matrix. Okay, and the next question is what kinds of summary statistics you can calculate from the B, the L, the M hat. That's estimates. Okay, then you can rank the summary statistics. Yeah, at the beginning, we think about whether we just use L2 long to do it or other long. And actually, I killed most of these kinds of ideas until we think we should do some using operating law. This is basically we choose the largest eigenvalues because I think that in most of SNPs, this B hat M just simply just noise. Okay, and if you're using L2 long to do it, therefore you have accumulated noise, and actually you can only detect the strong signals. That's the reason we think about it. We should not use L2, this L1 or whatever this type of the aggregation type of the summary statistics. That's the reason I talk about just choosing the largest eigenvalues, and that's the basic what we did. Okay, then we basically were doing rank 1 screening. That means if you most of beta ML is the 0 matrix, and we prefer to use the rank-based screenings. We find that this perform much better than L2, this type of the screening method. Okay, and this is basically after you're doing screening. And then we can introduce the nuclear alarm penalizations to do it. And actually, what we find is that this theory is not, for screening properties is pretty standard, okay? But actually, when you try to do it for operator, for the operation norms, that's make the things much harder. And this is the theory for estimation estimators. We have the estimation consistency and the rank consistency, and also convergence rate. Okay, and this is basically the unified theory for the two-step estimators. You can take a look at our paper in JASA. And this text is it's published 5 years ago. Okay. And this is the basic of the data analysis. We did adding data, 735 subjects, and each resolve these kinds of matrices. And then we're using the corrector for age, gender, and also SNPs, and the top 5 PCs. And then we identify the both right and the left on these kinds of chromosomes and the corresponding SNPs. Yeah, and we are able to identify ApoEs. That's the most common popular ones. We're knowing the kinds of SNPs for the late onset Alzheimer's disease. And this is basically the estimation results. And this is the one thing I did 8 years ago. And actually, this is the matrix treatment papers. And this paper is almost accepted by Johnson, but this is another also non-story. But this we finished in the last 3 years. What we did is we try to gradually move shifted to the doing causal inference or the mediation analysis. And the key message of here is we think about the Alzheimer's disease. Okay, the one left one is a healthy brain, the right one is Alzheimer's brain. And you think about what's the relationship between genetics and the imaging, and also the clinical cognitive impairment, and also disease. Okay, we try to move all the things together in certain degree. This is a pretty well known called AD pathogenesis by Jack J in 2010. These are hypothetical models of AD pathology. Basically you see that they thought about this genetic component kind of conversions at the beginning. Maybe you get it from parents, sisters, and then you start with this pathway is to have a temporal feature inside, pattern feature inside. Then it means your first start with abnormal A beta deposition. Then you have these kinds of abnormal total aggregations. Then you have this hippocampus atrophy. And to the behavior diffusivity, that at a certain degree, you have this diagnosis. Okay, you have the MD, MCI, or ADs. This is basically we try to at the beginning of the project, we think about how to really link all these things together. Okay, and this motivated us to do this kind of things. I think we started about 6-7 years ago. Just start thinking about this, because I'm in the last several years I'm working in the company. I didn't pay a lot of attention on doing this stuff, but we just need recently we started to work on it. And one big things we think about is how to do why don't we just doing some causal inference type of stuff or mediation analysis. Here we introduce a causal inference framework. Okay. The causal inference framework is, what we did here is the YZ is potential outcomes on the exposure levels, okay? And here, what is our exposure levels? Actually, we think about the hippocampus as a two-dimensional continuous treatment, okay? And that's basically our kinds of, we put this in potential outcome framework. The Z is a two-dimensional treatment, okay? It's the hippocampus surface matrix. YI is a potential outcome under the Z, okay? This YI is the exposure observed. It's cognitive impairment, this type of matrix. And actually, the aim is we try to identify effective surfaces. We want to estimate unconditional distribution of UZ as an expectation of the Y that's given Z. Okay. And actually, we are making these typical assumptions in causal potential outcome framework, and try to link all the things together. Okay, that's the key assumptions we made in the paper. And actually, the way we have this, actually, there are two equations in the model. One is basically Xi is all the set of correlates, scalar correlates, in particularly related to like the baseline characteristic plus the SNP data. The Zi is the hippocampus, okay, the shape. And this is basically, we started with the simply kinds of this functional linear models. But because the number of correlates is huge, and also the Zi is also a functional data. we are interested to estimate the effective surface. Okay, that's our target. And actually, we have this another models, these outcome generating models. This is related to Y. Then also we also try to model the treatment generating models. Okay, this is we assume that the Z is genetics also contributed to the hippocampus. Okay, and then you have the thing about this, basically the genetics contributed to hippocampus, and then also the genetics also directly contributed to the outcomes, the cognitive impairment. Then we basically have these two models merged together. One is outcome generating model, another treatment generating model. And then we basically try to select and the corrects, okay? Because you need to select correctors in the outcome generating models, and also you need to select corrects in the treatment generating models. Both are these two sets and may not be the same thing, okay? And therefore we clean up, we decompose the X, the corrects, into several key components, okay? And we decompose, you'll see that basically, and there's we have these confounders, XC, and also precision variables. What is called the precision variable? Precision variable means they only influence the Y, but it doesn't influence on the Z or other the treatment, okay? And actually we split, and also the XI is instrumental variables, okay? XC is a confounding variable and precision variables. That means we separate the correlates into the three partitions. One is instrumental variables, the only influence Z, and then one is only influence is confounding variables, another one precision variable. And here in this kind of XN is another set of these that have nothing related to all other variables. Okay, that's the basically XN is independent of DN1. Okay, and because of that, then we have much clean kind of partition of our covariance. Then after we finish that cleanup, and we can using these kinds of these kinds of two screening procedures to select relevant and kind of covariance. Then in order to reduce the data dimensions, the covariance dimensions. Basically, we combine outcome and the treatment models and select the submodels based on union of M1 star and M2 star hat. Okay, and this is the basic marginal screening based on both models, so that basically we minimize the missing some important covariance. And when you have done with that, then when you have the M hat kinds of include the two components, precision variables and instrumental variables, then you can refine estimate something like this, okay? And then you can get you can impose different penalty on that. And that's what we did. Then you're using L0 and L1 penalties. Here we're using L1, and also get these kinds of star or things. And then we apply it to our editing data. And for this very small number, 500 subjects is pretty small for us at this stage. But that's, I told you, that's the work we did almost 5 years ago. And what we did is this is the beta hat, this is the causal effects of estimation results, and the beta hat, okay. And this is basically the atrophy of CL1s, and the subiculiums cause the more severe the behavior deficit compared to CL2 and CL3 subregions, okay. And this is the work we did, our recent paper in JASA, okay. And another thing that we did is actually we also did some optimal minimax variable selection for larger scale matrix linear regression models. And we also did it by one of my collaborators. What we did here is we basically, this is a theoretical work. Okay. And that's the skill based on these matrix models. We try to work on it. And this is basically this is a selection procedures And this is basically the first of the procedure we try to do correlate selections and coefficient estimate, okay? And basically what we did is we screen out unimportant correlates and also to yield a consistent estimate of the coefficient matrix simultaneously and get a smooth estimate. The second procedures is basically we do sub-regions and detections, find out areas affected by predictors. Okay. And this is basically the correlate selection and coefficient estimators, and basically we use these kinds of the hardest thresholding correlation for doing that. Okay. And we do screening on the beta hat, okay? And this is basically the selected basically submodels, and this is the criteria we use. This is very similar to other papers on that. Okay. And this we can accept the sure screening properties and optimal minimax rate. And this is the paper published in the Journal of Machine Learning, and I think last year. And if you're interested, there's the theory method behind it, okay? And then this is the first step. The second step basically we do this sub-region detections, okay? And then we basically, we first the things, we are not just to estimate the correlates, the effects. Also, we are interested to basically on the images, we are basically decompose the images into at least two parts. One is inactive set and active set. And we try to detect the subregions with basically relatively active set, okay? What the X set look like. And then we basically try to recover this active set. We basically, that's the combination of the two methods about, okay? This is the last part of this decomposition-based canonical correlation analysis. Okay, what we try to do here is try to integrate the data across different domains. Okay, that's our key motivations. And you'll see that for TCGA, you have these kinds of mutation, copy numbers, gene expression, all the different omics data. Okay, from the same subject, okay? And for HCPs, you also have different images of data from different, from the same subject. And also then you have the different data type for the same subject. One question is, and how to perform kinds of data integration across different modalities. Okay, that's the one, the project we did almost 6 years ago, because one of my D9 students worked with me as a postdoc. That's the problem I threw to him. And basically, this is at the very beginning, we don't know how to do it, okay? And even right now, I don't think we know how to do it in comfortable way. We just try to solve these problems and to develop a set of methods. And this is basically the problems we try to do. Okay, you have this like suppose you have Y1 to YK. This is basically the different data types. Okay, and there's the set of variables. Okay, okay, this is the Y1. You have P1 variables. Then you have N subjects. You have also YK. You have the 1 to 2 PK. Then these all subjects. Okay. And you have multiple type of data sets from the same set of objects. And how do you do with this? Okay. And actually, we go to the, this is the only one approach we take. We started from statistical literature first. Okay. And in particular, my collaborators, Steve Merrill and other people, and they basically assume that they are decompose each data type into the 3 components. One, they think they call it common, okay? Kind of, another one is called distinctive, okay? That means they write YK equal to XK plus EK, okay? And they think that this is the CK okay, for each of the Ks, these C1 to Ck, capital K, have something in common. The D1 or the DK is something different, okay? That's the kinds of the data type of specific components inside, okay? And there's many papers published by using these models, and nothing wrong with that, okay? And when we started this project almost 6 years ago, what we did is, okay, and we think of the CK is called the shared underlying mechanisms of the K-COM data set. DK is distinctive resource matrices, okay? And then the key question is that goes back to how to define what is the shared kinds of the features across the modalities or different data type. Okay, and the different people have the different definitions. Okay, and the key things is this is the set of the names to jive, ijive, and ajive, all these on plus and the discos SCA and the COBE. This is all the different the kinds of the method that we developed for this type of decomposition models, and nothing wrong with that. Okay, the only this method is difference is not in how to define the CK and the DKs. Okay, and that's the important. Okay, and what's the problem associated with that? Okay, that's the project I did. And there's two major drawbacks. Okay, and they define all the CKs and the DKs on these kinds of Euclidean space. Okay. And it's not on this L02 space, okay? And the key things is this IN is approximate of these things, okay? And the key difference is they treat all these things in Euclidean space, okay? This IN, okay? And what we try to do is characterize these things is in L0 space in the sense of the set of all the random variables. Okay, with zero mean and the finite variance. Okay, and you will find that when they try to define it, they truly doing things for the metric space. And the way they define the correlations, the orthogonality is purely based on these two vectors are orthogonal to each other. Okay, from that aspect. But you will find that if we using this L0 squared space, when you, in certain degree, you define the orthogonalities by using the correlation structures. Okay, these two are different. Okay, these two are different. They are not the correlations. You call it zero. That's not the same as this orthogonalities. And the focus and all these existing methods focus on basically the CK orthogonal DKs, this not well considered the orthogonality among these DKs. Okay, these are the key problems associated, even for the two-dimensional things. Okay, these two vectors in the Rn space, if are orthogonal to each other, that means the inner product equal to zero, does not really infer this correlations. Okay. And that's the basically try to offer, they try to avoid the common nature factors between D1 and D2s, okay? And what we try to do is we change the space they belong to. We basically take the XK equal to CK plus DKs. We define CK and all the things from these kinds of the random variable space, okay? And then we try to basically doing the decompositions in the sense of the span of C1, the span of C2s are the same, the common native factors, and also uncorrelated the distinguished native variables by using these two spans orthogonal to each other by using correlations. Okay, then this is basically the key differences between our approach compared with all these metrics decomposition in the sense of we're working on the two different space. Okay. And so that the properties are different. Okay. And then we basically to have these latent structures and your C1 to see this stuff, and basically, and that's the shared components, that's the independent components, and how do we define these kinds of data structures decomposition for that? Okay, for the k equal to 2. Okay, and this is basically the geometric kinds of characteristics, how to define the orthogonalities, and why should we do that? And if you're interested, you can just take a look at our paper. And this is basically the key estimation procedures in the papers, basically just to tell you how to reconstruct the shared component and the distinguished components. The key difference is now in how do we really incorporate which space we should work on it. And actually, we already extended this one to the k greater than equal to 2 space data sets, and show how to do it. Okay, and I won't go to the details of the theory and also simulation studies, and that show you that the existing some problems. Our method, our current method is competed with the existing method. In what sense? Okay. Yeah, we just applied. This is some data. I will share the data analysis results slides with you guys, so that you can take a look at that. Let's take a rest for 5 minutes. We started at 4:10. Any questions? Any questions so far? Yeah, I think the previous lecture 4 is just to give you some examples. How do we do some extending some existing statistical method and to the neuroimaging data. But all this extension is quite standard for most of the statisticians. But we don't know. I think that at least for first several projects, quite natural, and basically that's the views on the existing methods. And for last several projects is harder because we don't know whether what we did is right. But I think that's a nice trial. But I think that there's another opportunities over there. And then because of the time limitation, I want to have the chance to go all to other topics, and I just pick up something to present. Okay, and the last part I'm going to teach here is about the imaging genetic analysis, and this is basically. And in any case, I'm going to upload all these slides to my website, and I will send it to the link to you so that you can download. And I'm going to share my review papers on this topic pretty soon, and I'm still try to finish to writing that. Okay. I think in the review papers you have a more deeper understanding of the different things that we have been doing. And why you can easily identify some interesting topics, big topics to work on. And to me, there's tons of opportunities for doing that. You think about these days, you think about the data integration across different domains, different data type, and the imaging genomics, and also this lifestyle data, like nutrition type of the things. They are all the major data types in the field for the medical science. And I think you have to, in a certain degree, if you really care about applications, they are all the kinds of skills you need to have in order to really try to model this type of data. Okay. And I think the last part we try to talk about imaging genetics analysis. And this is still an emerging field. And there's a lot of development around 2012 to 2018, something like that. And but at the beginning, the statisticians or these other people jump inside to try to do some modeling. Okay. And I still feel the one way is all these existing methods really not good enough, and just it's nice to try. Okay, basically the key reason is when we started to do this topic, and the number of subjects in all the imaging studies are pretty small, because most of the data, largest data we have been handling is around 1,000 subjects. That's the reason, and also that's the reason in many cases, the model we developed so far, you will see there's a lot of models we developed, so it's not really ready for larger scale, the larger number of subjects, okay? And even from this is a group of the papers published by my group, and also some of my collaborators, close collaborators. You will see that in the recent 5-6 years, I have been basically analyzed the imaging genetic data from around 50,000 of images and the subjects. That's basically, I focus on processing the data and write up these clinical findings in the last 5-6 years. We have a series of papers published in top journals, scientific journals. Okay, and I think that one reason is I slowed down the method development because I think that I changed it. I take another trick to do it. First, I process the data and write it up, And for all the imaging data, I didn't really work on this all the I did the data dimension reduction first, okay, and concentrate a relative large number of the phenotype, maybe several thousands of phenotypes for the structure images, function images, and DTI data. And I just want to see these kinds of the small set of the number of the phenotype, whether this, because this phenotype usually is pretty robust and is basically is reproducible. And I want to see for this set of the signals, the phenotypes, whether we could understand their genetic architectures or not. And amazingly, basically, for all these structure MRI, diffusion tensor image, and the function fMRI, even for diffusion, the function fMRI, we're able to identify, to understand the genetic architectures, all these brain white matter and gray matters, okay? And that's the reason we have a series of papers published in Nature Genetics and Nature Science. And actually, we have some follow-up on that direction. Nowadays, because we run through basically, the trick we take is basically we process all data by ourselves. And then we basically doing data dimension reduction to focus on some of the major signals for this, for brain function structures, and try to establish their genetic architectures. And now we have the first level analysis, understanding of these problems. Now we are, and when the more things that we have been working on, and we find that we need to redo all these methods. And we find a lot of problems with this. And it's not the papers published in the field. There's all these kinds of, well, I think you will see a list of the follow-up on this topic, because from the method side, okay, we are trying to, the more data analysis we did, and we understand the problem better, and then we motivate us to do another set of tools. And I think this is just some reference to the papers from our own publications. If you're interested, you can take a look at them and see what we did. And what's the motivation for doing things? And the key things is for imaging genetics. What are the key things? Why do we care about that? And the key things is for most of the brain-related disorders, like psychiatric disorders, like schizophrenia, autism, and all these Alzheimer's disease. They are very complex traits, and basically genetic account for majority of variation, a large portion of the variation for this complex disease, okay? And when we try to do the imaging genetics, the real key reason is imaging genetics allows for identifying how common a real genetic polymorphism influence the molecular process, binds the neural pathways, and mediating individual differences in complex behavior process, related disorders in responding environment to adversaries, okay? And for the neuroimaging phenotype, so far what we discussed for the structure MRI, function MRI, and the diffusion tensor images. And most of these features are object, okay? Either is a functional data, matrix data, or like a manifold value data, or the network, okay? This is the only different data types. And then the dimension is varied from 100 to like 500,000, something like that. And actually there's a series of papers published by my collaborators. They find that the brain structure is highly heritable, in particularly like the whole brain volumes and the volumes for some structures, okay? And this is basically these days we were using we started with from the SNP data, and actually the UK biobank data set and has this the exon sequence data, and also they are going to release the whole genome sequence data for that. You know, we haven't tried to do that much. We only finished that data analysis for the SNP data, okay, for the Zuckerberg studies, for these imaging traits, okay. And at the very beginning, as I said, for the imaging phenotype, we are only work on these kinds of a small set of the imaging phenotypes. Okay, basically we take some dimension reduction methods and try to perform that. Okay, and you will see the more kinds of follow-up papers on that, but that's not the case in this lecture. And the imaging phenotype, we know that is extremely spatial correlated and high dimensional, and basically they have the strongest spatial smoothness inside. And this genetic data, you also know that the genetic data is also high dimensional and correlated. Then usually you can cluster snippet Smith into these local blocks, and basically, and you can create this called LD blocks. Okay, and this the imaging genetic studies. This really depends on the type of imaging phenotype you're working. And one set of this work is done by the Ann Brown's Institute. Okay, they are not just Ann Brown and the Institute really looking at the imaging genetic brain from the neuron levels. Okay, they create a lot of kinds of the imaging data at the neuron levels. Okay, that's the cutting edge research on this topic, in particular related to that. Okay, and they don't do too much on the neuroimaging data, but actually they have single cell and all this type of data, all the gene expression data, and the RNA the impression on that. And that's another basically the imaging genetic study, not just about white matter or gray matter, these kind of things. And actually the GBM, the brain cancers, okay, this is also is very interesting in the sense of we also, for like a TCGF, we have this H&E and also some of the CT scans. And we also can scan their tumors, okay? And also could have gene impression data, and we can correlate this kinds of the genetic or genomics information with the imaging phenotype extracted from the CT scans or other kinds of the tumor, okay, information in the tumor, like the radiomics information. And the adenine is one kind of the first project, the studies we started to work on the integrated imaging genetics. We started almost 12 years ago and basically try to develop the method. And we didn't move very fast, okay, just because the data set is pretty small. And we just focus on just to cook some papers, and we doesn't see a lot of, we didn't produce a lot of impressive results, many because the data set itself it's not very promising at that time. But we did start earlier than others. Then until the certain stages, then we have the UK Bank data set, and we can move much faster than that. Okay, that's the basic opportunities. We took advantage of that. Okay, and actually NDAR you also have shared the imaging genetics data in the public domain, but nowadays we have around 100,000 subjects with both imaging and genetic data. We keep on basically assemble more studies with both imaging and genetic components on to create a large database. Okay, and this is basically some of these original analysis. This is just the single SNP with the single quantitative traits, and basically doing these kinds of association analysis. Okay, and if you read one of my friends, Shen Yi and Paul Thompson, they wrote a paper about imaging genetics. The method that they are using in that papers, many using regulated periodization method. But then when they do that, they already basically reduce the data dimensions. Okay, then so that you can run the regularized method to do that. Okay. And actually, another thing is in the history of the imaging genetics. The number one is Adam. Okay, number two is Enigma. Okay, this Enigma was run by my friend Paul Thompson. He basically he ran the kinds of international effort from around 200 institutes with more than 300 co-authors worldwide. They formed these consortiums. Okay. And what they did is basically it's more like federation analysis or mediation. They essentially doing meta-analysis. They basically each side contribute a set of the data set, and then they run all these imaging process pipeline and also imaging GWAS analysis locally. Then they can integrate all the data results together to merge all these findings by using meta-analysis. Okay, that's the way they did it in last almost 12 years. They expanded basically to the different kinds of psychiatric disease and also Alzheimer's disease. That's what my friend Paul Thompson did, and he expanded to multiple countries on this project. And what they did is basically the meta analysis. Basically, they developed the imaging protocols, also developed the statistical protocols, basically how to integrate the data, the process data consistently, and also try to run this meta analysis across the different sites. And this is the basic thing how to do these kinds of data analysis on that. You will see these kinds of, they publish several papers in top journals on Science and Nature, all this Nature Genetics, Nature Neuroscience on that. And this is just show you one Enigma's findings, and they published in the Nature Genetics in 2012. And there's the pioneer project, the hippocampus intracranial volumes GWAS meta-analysis. Okay. And this is they based on almost 20,000 of the individuals in discovery plus the replications. That's what you think about it, almost 10 years ago. It's a huge sample size. Okay. You think about before that, all these imaging studies, they have less than 1,000 subjects. Not talking about imaging genetics. Okay. Another one is genetics of the subcortical substructures. And basically the discovery and the replications about 30,000 individuals. Okay, the 50 contributing cohorts around the world. Okay, and they basically define some consistent findings in that. And this is the the meta-analysis they did, and they get the findings associated with it. And this is another paper published in Nature Genetics is generalized meta-analysis identify neuroscience and the functional pathways influence Alzheimer's disease risks. Okay, basically, how do they do it? This is basically and try to integrate part of the data from the UK Biobank and to do that. But this one is mainly it's not for the imaging phenotype. This is mainly related to the clinical phenotypes by using UK Biobank, ADSP, IGAP, and the PGC ALF2. Okay. And what's next for Enigma? What they did is the GWAS of the cortical areas and the thickness. They assume this 30,000 subjects. That's basically what they did in the last several years. But this nowadays the sample size is much larger than this. They because of the UK Biobank. Okay. And this is the Enigma DTI data set, and basically they run this DTI analysis GWAS, and for multiple sites. Okay, and they didn't see a lot of strong signals for their data set. It's mainly because the data is not the data is very heterogeneous, and data quality is not that great, and that's the reason they won't be able to find many interesting results in their DTI GWAS analysis. Okay. And also they did this kinds of called connection-wise and genome-wise the analysis. You're basically using the extract the resting function of fMRI connection or DTI connection results as a phenotype, and then run the GWAS on that, and to do this called the connection-wise, genome-wise screening these kinds of Alzheimer's risk genes, and they're able to identify some well-known genes, kinds of Alzheimer's disease. And this recently people are thinking about how to integrate this polygenic risk scores and in the downstream analysis. And basically the idea is this is very become popular these days. Okay, the key ideas is basically you're doing summary statistics from independent GWAS on a base phenotype like case control or like intelligence, this type of continuous or binary dichotomous traits. Okay, and then you're basically doing like something like the SNP associated with the base phenotype. Use the user specified P threshold, or you can take all these, and then you index this SNP like LD based clumping to select the most significant SNPs from each clumped regions to focus only on the independent signals. Then you combine these things to get this effect size of indexed SNP. Then you can dump this study to these kinds of for the studied imaging genetics cohort, and because you have individual SNP data and you also have targeted the phenotype. These targeted phenotype may not be different from this summary statistics because that usually like Alzheimer's case controls. Okay, that's based on that genes. But we are, you can combine these kinds of PIS with the candidate PIS, and basically add these together, you get these kinds of predicted values. Okay, and optionally you calculate set of the candidate PIS and pick the best ones, use rigorous strategies, you get a final PIS. Okay, and then you can add these kind of imaging phenotype. Basically, you can calculate imaging phenotypes, the PIS effects on the imaging phenotype. And that's basically what we also did something similar on that. This basically the PIS approach for doing that. And then how do you mapping the connectivities? So basically, you can use structure MI, you can use diffusion tensor images, and together to do it. And this is basically the papers they published in Nature. Basically, it's the GWAS association as the brain imaging, the phenotypes, and on that. Yeah, this is basically the first imaging genetic paper from the UK Biobank, because with my friend Stephen Smith from Oxford, when he published these papers, then in that case they used about 8,000, less than 10,000, 8,000 of the subjects from UK Biobank from phase 1. Okay. And they generate about 3,000 of imaging phenotypes. And they are basically run through these kinds of these 3,000 of different GWAS and on this data set. Okay. The 3-dimensional 3,000 of imaging phenotypes and also about 6 million of the genetics SNPs. Then they run these kinds of this number of the GWAS analysis, and they get these kinds of findings related to function fMRI, DTI, all of these things, structure connectivities, all the things. They did these kinds of the pheno-wise, genome-wise association analysis. Okay, but I think when they then they also have a follow-up paper on natural neuroscience. They do and found the X chromosomes. But actually, when we try to replicate and also try to do all these things, and we find, we did something better than what they did. Okay, that's the reasons we can do. We have a lot of new findings. And the big brain imaging genomic cohort, and our group has been systematically connecting public available individual level data for over 50,000 individuals to build the largest database. Now this number should be 100,000 individuals. And we try to incorporate all these kinds of imaging genetic data from the literally public available resources to build such a database. And actually, if you're interested, what we did, and for the brain imaging knowledge portals, this is our website, BigKP.org, and we aim to build the basic knowledge basis of neuroimaging genetics. If you're interested, you can just go through that. I also have the summary statistics shared. You can download our summary statistical results. Okay. And this is basically the key motivation of doing these things. And we are trying to move to the second wave of the method development. We're changing the whole areas in a certain degree. You will see that in the next few years. And the overview of the topic, this is basically you think about the whole areas. Basically, we have environmental genetics, brain imaging, and also cognitive behavior, and also the diagnosis, and also the design related to selection of the samples. Okay, and you think about we have all these data together. Okay, for imaging genetics, and we mainly think about we're trying to build the pathway from the genetic to the brain. Okay, this pathway, and also we try to link. You see that another things that we're working on is genetic, the brain, and also the cognitive or diagnosis. Okay, and we try to basically link all these things together and think about from big pictures. Okay, and we wanted to build as I said, in the last several years, we have to be working on build these genetic architectures of the brain structure and the function. Okay, and the common goal is basically detect the potential genes for inheritable phenotypes. Okay. And this is the brain in genetics learning problems. My friend classified the problem into the 3 categories. The A is learning problems. And basically the heritability estimation, imaging genomics association, and also integrated imaging and genomics for outcome predictions. The second one is biomedical applications consideration. The third one is the statistical and the machine learning considerations. Yeah. And you can read these review papers and basically get a sense of that. Yeah, no, I think you can do better than this. Okay, and this is basically fundamentally we have this SNP level GWAS. We also have the gene level GWAS, and the C is multi-array expressions, and 2 gene to RNA expression matrices. And the D is gene set of the brain circuitry modules. And then you can merge these 2 together. You get the imaging genetic enrichment analysis and do annotations. This is basically the fundamental from the pathway as a network analysis, how to really integrate the imaging and genetics together and how to do it. Okay. And for statistical method, you have the different level of genetic informations, and also have a different level of the ROIs related to imaging. Okay, and what we did is in the last several years, we basically focused on the candidate ROIs, and we ran through all these GWAS data. Okay, as much as we could. Okay, and basically we got all these clinical findings from that level. And we are just pushed to the next level for many ROIs and works-wise. And that's basically, and try to complete the whole big pictures about that and to see how much we really can get for that. And I just show you something what we did. That's the work we did 10 years ago. I just we still think about how to refine all these tools we developed. And I just show you some key ideas. The first thing this project we did is like Y is a phenotype, X, beta, and plus E. And here is the PY, the phenotype dimension is high, the dimension of phenotype is high, and also the dimension of the genotype is also very high. Okay, how do we do it? Okay, and this is basically, that's the current approach, like candidate genes, candidate imaging regions, and basically you are only looking at the particle features, and then you can do the genome-wise and voxel-wise analysis, but the statistical power is low because you need to basically account for a lot of the searching space. And everything in the big models plus pillar method, and actually you know that the variable selection is not really, it's not designed for this type of problems, okay? Because the variable selection, usually you have to sparse signals and to do it, okay? Although if we don't have sparse signals, then the current method needs some modification for doing that. But in general, for this large scale data, I don't think there's a single, everything in a single model is a good way to handle the big data. That's simply impossible, okay? And the model for candidate RY and this stuff, that's the pretty relatively straightforward. And basically you have the phenotype, and we just do a high dimensional regression models, and you have a genotype, and the key things is the dimension of the genotype, and also the dimension of phenotype are pretty large. And we can impose the certain degree of the certain assumptions, like sparsity of B and the restricted null space property of design matrices. And the one thing we can impose on the B is we did the two things. One we did is the no rank structures on the B. It's similar to the factor models, and plus the sparsity assumptions. I think the Jensen did some theory on this. They call this the integrated this factor models plus the sparsity. Basically, they put together, then you capture majority of variation inside. Then we can impose the assumption on that, okay? And you can use regulation, all kinds of regulation methods to do it, convex, non-convex, your regulation periodic terms to do all these things, okay? You can do a lot of different things on that. And then you can calculate the genetic or imaging networks, and by using the B matrices, and you can calculate. And this is basically what my friend did is they basically show that by doing that, basically the proposed method is performed simple multivariate voxel-wise, genome-wise association analysis. And then you can do these SNPs to genes to pathways, and basically in order to incorporate the pathways, basically you can group the genes together to base on the existing pathway data. And also you sometimes you see these overlap pathways, and how to and then you have these kinds of the group to the regression coefficients, the sharing across different pathways, and how to deal with that. You add an additional penalty of that. And this is basically what they did. And this is basically the AD and the normal controls. And this is the demographic informations. And they can get to the mean and standard deviation of the SNPs, and also the p-values, minus SNP and p-values. Okay. And also you can do this map the SNPs to pathway, and then you can reduce the data dimensions. And like you can use KEGG to get this pathway and analysis. And you can do some of these screening methods, then you can calculate p-values on that. Okay. And this top SNP and the gene ranked by the method. And another thing that you did is, besides your impose structure on the B, and then another term related to the error terms E. And what we did is similar to what the other papers. Basically, we impose long-range correlations plus the short-term and correlations. Okay, that we did the similar things on that. Okay, and in order to reduce the dimension of the sigma e's, and these are the different kinds of the patterns for the correlation patterns we did. The first column is the different coefficient images. Then this is the other things, these occurrence metrics we did. Okay, and then we basically compare the sensitivity or specificities, and for different methods that we did, and we are able to do it at the ROI levels. Okay, and then we replicate the 40 candidate genes on the Alzheimer's website. This is basically the ROI network and the gene networks we created. And the other models for the candidate SNP and the genes voxel-wise analysis. And for the candidate SNPs, that's this is basically this is the results almost 12 years ago, and they're pretty simple at the beginning. Okay, these are the models we focus on. It's still these models here, but the difference is the PY is much larger than sample size. This N is much larger than and PX. That's basically we focus on gene set. Okay, and then we take this kinds of mixed effect model approach and to do it similar to the existing literatures, and we just replicate whatever the people did for the one-dimensional case, and then we extend it to the high dimensions. Okay, and then we can do this RAMO stuff, weighted RAMO, and to incorporate the spatial information smoothness inside. And that's basically we just did some homework to do that. Okay. The key differences between our method and the existing method in the genetic literatures is basically our Y is continuous variables, or just we have functional data, and how do we deal with that? Okay, and we basically constructed this weighted REML method. Okay, then you can calculate the hypothesis testing, get the p-values, then you try to improve the powers. Okay, and also we can construct the score statistics, and this is basically how do we do it, this stuff. And also we constructed this weighted score testing statistics. Basically, we are incorporate the smoothness across the neighboring voxels and get this weighted score testing statistics. Okay, and then if we just direct calculate this takes a long time to calculate it, but we have some smooth trick to do that. And then we can do hypothesis testings. And basically, the first column is a voxel-based analysis. The second column is the true values. Then the third column based on our method, based on by integrating the smooth properties. And this is the basic statistical power for doing that. Okay, you see that our results is much powerful than the existing method without considering the smoothness. Okay, and this is basically the avenue results we did. And by using almost 335 and all the imaging data we have generated. And this is basically the 45 hours by our method. This is basically the findings from this analysis. And also the model for candidate ROIs, whole genome wise analysis, okay? And for this one, then this is where we have these kinds of the PYs, even though it's bigger than N, but it's not that big. And the current approach is if the dimension Y is not that much higher than the sample size, what can we do with that? Okay, the current approach is to treat it as multivariate responses and try to correlate it with covariates. And you're basically doing like a multivariate linear regression plus Cotillion t-test. And or you can using the PIS, principal component regression, to do it. Or you can do the component-wise method to do that. And also partial least square estimate regression to do it. And there's all these different limitations associated with all each of these methods. And this is the basic, what we did is, and we consider these sparse projection regression models, y equal to b to xi plus e, okay? We are interested in the c to b equal to this. How do we do this stuff, okay? And what we did is we try to project a yi into some space. The w is latent space, okay? And basically, like we project in this space, and so that the w transpose yi is very low dimension, 1 or the 2, okay? And then we can reduce this hypothesis testing probably into this. And the question is how to determine the w, okay? And actually, then you can using this generalized attributor those ratios to do that. And it turns out the ratio of the two kinds of the quadratic forms. And how to, if you the sigma r, the dimension is so high, and how to deal with it. And there's basically one approach is you impose the penalty on it, okay, sparsity on it. Another thing is you can split the data into the one is estimation, one is testing, so that basically you don't get into the big issues related to hypothesis, the estimation. And then you can do the t-statistic on that, okay? And then you can calculate the p-values by using wild bootstrap. These are the papers we published in JAWS maybe 10 years ago, something like that. I don't remember. And then we basically test the different settings, simulation settings. You see that our method perform very well on this relatively, like the dimension y is relatively close to sample size, okay? And another thing that we work on is like a functional voxel-wise generalized analysis. Basically, we really treated the data as a functional data, okay? How to do it, okay? Only problems when you work on the extremely high dimensional phenotypes. And because the sample size is getting bigger, the number of kinds of the points on the imaging data become bigger. So then also you have the high dimensional stable data. And then number of the tests is extremely high, okay? That makes this almost impossible to do it because given the number of the size of the searching space. And this is basically, typically the people do this kind of stuff. And, but they find that this method is not a very impressive because they use 500 clusters run month and they get to the final p-values, very non-significant. And this is making this basically that's almost the work they did 10, 15 years ago. And they find that this method is not that impressive. And we cannot find a lot of impressive signals in the data set. There's a lot of false positive inside, okay? And what we did is basically we tried to incorporate the spatial and temporal the smoothness inside and to increase the statistical powers, okay? And what we did is that we're still doing a similar thing to our variance coefficient models, so we can develop this water testing statistics. And the key things is we have to deal with is extremely high dimensional X and also high dimensional Y, okay? And then make the computational time is too large, you know, to be practical. And actually what we did is we developed some screening procedures to reduce the data on both sides. And in order to, at least for the imaging genetic side, okay? And this is basically we tried to come up with a different way to calculate the testing statistics in order to speed up the calculations. And this is what we did is we doing these global sure independent screening procedures to reduce the imaging genetic dimensions and and tend to the detection procedures at the end. Otherwise, it's almost impossible to run this data. You think about it, you run this kind of GWAS for millions of the phenotypes. It is also a disaster for doing that. Okay, and this is basically the key features. How do we achieve the kind of the dimension reduction on that? Okay. If you're interested, you can read our papers. They show there are the global show independent screening procedures. How do we reduce the computational complexity of doing that? Yeah, our computational time is on single CPUs. That's about this many seconds for doing that. Okay. On the same data type, data set, you see that. Let me see. Okay, there's another things we did is, I think this method is quite interesting. And why this is interesting? Because there's the one things, you know that for diffusion weighted images, we can extract a lot of white matter tracks. Okay, then this is basically the, let me see. This is the key point, because in the brain we have the many pair of the white matter tracks. Okay, and you can run these GWAS for each tracks. Okay, this text and you combine all these GWAS results together. Okay, and basically you have the functions defined in the different locations. One thing is when you have phenotype is large. Okay, how do you really do it? Could you do something different? Okay, that's what we did. I think this is pretty smart ideas we did for this. That means basically, and there are several challenges. The statistical inference on the variant coefficient is time consuming, and also analysis based on single thread is not powerful enough to detect the signal on the global scales. And the different fiber bundles sample are different coordinates, and the potential intercorrelation between the bundle statistics should be properly modeled. Okay, this is basically, and what we did in model is we wanted to perform joint analysis for multiple or larger number of the fiber bundles. And also we will try to take into account the functional spatial features and potential between the track correlations with the data. Okay, and also achieve the fast and efficient basic computations. And we developed this method called these kinds of hierarchical functional PCA regression models. And basically what we did is we extract the features for each bundle, okay? And after you've done that, and then we run the PCAs. You see that after we've done that, basically we run these kinds of PCA component on that. Basically it's a kind of hierarchical modeling, okay, for large number of the fiber bundles in the space. And then we merge all these features together, run a latent factor models on that. So that these kind of latent factor models allow us to decompose the data into the common features and the independent features. And then we basically to do these kinds of the fittings. Then we can construct testing statistics on that, okay? And these are the kinds of theoretical results we did, okay? And just to show you what we did for this real data. And we have these 40 fiber bundles, okay? And then we basically, we include this, we run these models for this kind of, we generate the data, okay? These are the simulations. We have these kinds of we simulate the common age effect shared by multiple traits and testing the significance of age effects. And this is basically this is the first, the top set is our new method. The following the set is our original method based on individual tracks, okay? And you'll see that if you're interested in the total tracking effect, and you'll see that we can increase the statistical power for doing that. And this is basically expanding how the proportion of the tracks with the true effect influence the performance. And this is we apply to our real data. You see that when we run this for each track, we have 40 tracks. Each track we run with extract the 5 functional PCs scores. Then we do factor analysis on that. And actually this is basically the number of variation they are counted for our factor scores. And this is basically the heritabilities for our each of the tracks across. And then this is the GWAS results associated with that. Okay, we didn't run this stuff. This is the paper did almost 8 or 9 years ago. We didn't run this on the UK Biobank. This is what we are doing right now. We tried to apply our tools we previously developed to the UK Biobank, see what's going on with this. Okay, but that's not our ongoing research. We didn't report that. Okay, and this is the last part. I just use 3 minutes to wrap it up. Another, I think, interesting things did by my students is a powerful global test for functional phenotypes. And this is basically the problem of interest. We have rank coefficient models, and we have univariate rank functional global testing statistics. The current method is basically using like the word type of statistics you weighted across the tracks. Okay, and or you can using the functional type of the testing statistics to it, and for functional linear model, this is did by my friend, and is there any way to make these procedures more better, more powerful? And because you know that when you do these kinds of you weight it across all these points on the function, you are losing a lot of statistical power doing that, because if the majority of signals are pretty noisy and zeros. This is basically to show you that one idea. So if you think about effect size is something pattern like this, okay, majority are zeros and only a small portion of regions have effects. But if we have something like this, what you can do about it? And also signal at the individual level, signal to noise ratio is pretty low. Here is only 0.35. Okay, how do you do it? Okay, and one way we did is just similar to the paper we did is we tried to introduce a W as a smoothly weighted function along the curves. Okay, we project our data in the low dimensions and take the inner product. Okay, and then the problem is how to determine this WS. Okay, and actually, and we still try to do this optimal projection directions. Okay, and actually we try to wrap this paper. Okay, and this is the basic. We find that if we do that, and this is a more powerful function of global testing statistics on that. Okay, and actually we did a simulation study to show you this is the beta on the arterial hypothesis. The signal to noise ratio is around 0.35. And this is basically the type 1 and also the powers. Okay, and you see that the conditional, the water type of statistics by aggregating the local testing statistics essentially have no power to detect anything. Okay, locally, globally. And our method is much powerful than that. And you'll see that this is the basic type 1 of all the powers we did. And we did run this analysis on UK Biobank. We did. I don't remember that. Okay. And actually, this fMRI we did on this the data from the UK Biobank. And this is the wild bootstrap. This is QQ plot, the wide bootstrap statistics. And that's the findings from UK Biobank US results by using our method on the track. The top 10 variation from the GWAS. And this is basically just to give you some set of the kinds of results on the imaging genetics. And we are trying to move this field much forward these days. And actually, I'm going to post all my PowerPoint files on my website, and I will send the email to you guys so that you can download. Any questions? Any questions?
Okay, if there is no question, thank you, Professor, for giving the short course, and thank you to the audience for attending this event. Okay, I think we can conclude and close this webinar.
Thank you.
Thank you, everyone. Have a good day.
When are you going to send me the link for my videos?
Oh, I'm sorry. Sorry, I didn't catch you.
No, are you going to send me the videos, the link to videos?
The videos, I think they will post the recording to the Hova. Do you have access to the Hova?
What's that?
Oh, it's where the there's a app called Hova that you can access all the sessions for the
Okay.
Right. I'm not sure if they sent you an email to give you access to the Hova.
Yeah, I know. I have that.
Oh, okay. Then you can find the recording on there.
Okay. Thank you.
Okay. Thank you. Bye, Professor.